opportunity to present today's program. I would like to welcome you all to the special lecture series hosted by the Department of Studies in Genetics and Genomics, University of Mysore. I welcome our beloved chairperson, Professor S.S. Malini, faculty members, research scholars and students. I would like to welcome our esteemed speaker, Dr.T.Shivanandappa, who is here to deliver the lecture on evolution of genes, form and function. Hearty welcome to you, sir. Let us commence today's event by seeking the blessings of Almighty. I request Sambura to present the invocation song. Thank you, Sambura. Now I request the dignitaries on the dais to officially inaugurate the event by lighting the lamps.
Students, class rep.
ಜ್ಯೋತಿ ಬೆಳಗಿ ಬರಲಿ, ಜ್ಯೋತಿ ಬೆಳಗಿ ಬರಲಿ, ಅರುಣೋದಯದ ಸೂರ್ಯನ ತೆರದಿ ಮಂಗಳಮಯವಿರಲಿ. ಜ್ಯೋತಿ ಬೆಳಗಿ ಬರಲಿ, ಜ್ಯೋತಿ ಬೆಳಗಿ ಬರಲಿ, ಜ್ಯೋತಿ ಬೆಳಗಿ ಬರಲಿ, ಜ್ಯೋತಿ ಬೆಳಗಿ ಬರಲಿ.
Thank you, everyone. Now I request our chairperson, Professor S.S. Malini, to address the gathering.
Good morning, everyone. A warm welcome to lecture series, which will commence from today. And every Saturday, we are going to have a feast of knowledge from competitive authority in different domains. Heartily welcome to all of you. Speaker, today's guest speaker, Professor Shivanandappa. He is famous as, he is known as ever-young gentleman. Okay, he never ages, and if anybody wants to do research on him, you are most welcome. Don't ask his age. And no, he is an inspiration for everyone, and always he will be busy. Till now, after his retirement, he was a emeritus professor in the zoology department. He used to attend conferences, publishing paper, and all of a sudden he will be absconded, that in African forest, Amazon forest, Dondeli forest, doing photography. And he is a great orator, writer, photographer. More than that, very jovial person. So for me, he is a stress buster. If I am very stressed, no, so I keep conversing with him, and he will make us always happy. Always he will be cool, happy, and that's a secret of his that's aging. Okay, he has not aged. Okay. So, heartily welcome to you, sir. I welcome all of you, all the research scholars and students, and this program will be for you people, and you have to attend. It is compulsory. Okay. So, afternoon I message you. There's an exhibition in a naturopathy hospital. Lot of exhibits are there. All of you have to go there, and everyone has to report me about one exhibit. It is compulsory, and attendance will be taken for all the seminars, and now it is getting ready. And if you don't attend, you will see Kantara 2. It has not released. I will show you what is that. Okay. So, because all these things we are doing for you people with lot of efforts, and with these few words, I welcome sir, and I request Amol to introduce sir to August gathering. Welcome, Amol.
Thank you, ma'am, for this great opportunity of welcoming such a great person. Good day to everyone. First of all, myself Amol Shinde, second year MSc, and I will take a great honor in introducing the guest of honor of our first lecture, that is Dr.T.Shivanandappa. I can assure you, all you young souls, that he is going to inspire you with his knowledge and experience, as he has been a former scientist, and with his contribution to with over 80 research papers. Dr. Shivanandappa completed his bachelor's, master's as well as PhD in the same great university you are studying it, in the field of reproductive biology. His contributions to the field of science is very much, among which I would like to mention one of his paper which intrigued me kind of. He with his colleagues has isolated and characterized a long-living strain of Drosophila, the Drosophila which we consider the central of genetics. He has been a former member of many scientific communities such as Society of Biological Chemistry, Society of Reproductive Biology, and Comparative Endocrinology. With this short introduction, I would like to hand over the mic to
Thank you for introducing me to the students. This is a place which I always love to come for many reasons. One of which is, of course, Dr. Mahadeshwara, who is my good friend. And Mahadeshwara's boss also is my good friend. And I mean, was not now, past when she was a PhD student. And as you all know, genetics is a department that took its birth in the Department of Zoology, of which I am a product. Those are the days when, the hallowed place of Mysore city was home to the brightest of teachers and students. That's why we're just inquiring from the students, because I'm seeing most of them are from Mysore, except a couple of them, few of them also. Those are the days Mysore students, I'll spend a couple of minutes just to make you relax before we go serious talking about science. Those are the days when Mysore college students were not able to get into the manasagangotri campus to take a course in MSc. Even when Dr. Mahadeshwara was a student, it was the same case. Getting an MSc was an achievement. And therefore, the students of Mysore were always minorities. If I am seeing a lot of students from urologist college. When I was a student, there were just 2 students from urologist college. Just 2. Rest of them were from all over Karnataka. So why I am saying this is, master's degree in any branch of science was a prestige. To do it in manasagangotri, it was not offered elsewhere. And genetics is one of the offshoots that emanated from the zoology department as it grew in specialization. And Professor Ramnath and Professor Ramchandra have now brought into an excellent stage wherein students are offered both PhD and MSc in genetics. So, genetics is one of the places which has a historical origin in Mysore University for a long time, ever since it started, because the founder of zoology department was geneticist, Professor Mahadeshwara, who himself was a student of Dr.D.R.Mahadeshwara. I'm sure you all know who is Dr.D.R.Mahadeshwara, right? Okay. Anyway, that's about my connection, because I was a student of Mahadeshwara University, and those are the days when we used to have great seminars, and the seminar was a means of great exposure to speakers, and even students used to take it as almost a challenge to give a seminar talk. I'm sure when already did the MSc also it was the same, not different. So today, activities like inviting people from outside is something very good exposure to students. And Professor Maurani, who is a very dynamic lady, some people call her dynamite lady. She is extremely active, academically very dynamic. Therefore, when she calls me for anything, not just doctor, I'm always there. So because we enjoy such bond. And she is the one who organized a national event from this department after she took over, which was the meeting of the society, it's a big conference. I'm sure this is one of the achievements after she came over to this department. She continues to be very active in promoting the cause of education, science, genetics, and zoology. So therefore, with this brief background, while I thank Dr. Mahadeshwara for inviting me, it's always a pleasure to talk to students. The reason is, talking to students is the best way to learn how to talk, best way to teach. So I am not come here as an expert, I am going to talk to you as someone who is very deeply interested in biology. And I am going to talk to you about something on which I am not an expert. I chose this topic because you are all students of genetics. I thought the best way to appeal to the enthusiastic students is to talk in a field in which they have chosen, they are interested. That is genetics and genomics. I'm sure you all know that is the in thing today, that genetics, genomics is very, very active area of research in biology. And you have all heard that 21st century belongs to biologists. And molecular biology, genetics have pervaded every field, from anthropology to medicine, agriculture to fundamental medicine, where the application of molecular biology, tools of molecular biology, are affecting the human lives in a very positive way. And greatest developments of science in the field of biology are not without the tools of molecular biology, everywhere, no matter what field you take. So it is molecular biology which gives those powerful tools for us to understand even a phenomenon like evolution. If you remember today, if you remember last year's Nobel Prize, anybody can name? Except research scholars, I expect you to not chairman. Anybody can name last year's Nobel laureate? No. sure some of you should remember last year's Nobel Prize. I am asking you just because it's not a Nobel Prize which makes a huge impact every year. Last year's is something special. Yes, who is that? Hey, in research scholar, Bala Bhagavanta Gautam Nanake. He has already heard a talk. Students are important. So, Nobel Prize winner was Svante Pabo. His work was on human evolution. He was responsible to reconstruct the story of human evolution, of the ancestors of human species today, Homo sapiens. And he sequenced the genome of the extinct Homo sapiens, because all that we had was either a bone or a skull. And from that we reconstructed the story of our human ancestors. And we want to know where we came from, who our ancestors were, what is our story on earth. So this reconstruction of human evolution was possible by genomic sequence that was got from a fossil. That was how the powerful tools of molecular biology helped us to understand how far our ancestors were related to us, how many ancestors were there. Now, through genetic sequence around days, we have discovered the human ancestor with just one finger bone. Can you tell me? Here are they. It doesn't matter. This talk is not on human evolution. They just to enthuse you that how powerful. And Nobel Prize in Medicine is not given for evolution. But it helped us to understand what are we, where we came from. And we have inherited genes that can either protect us against diseases or can give us new diseases. That's what Neanderthal genome told us. There is nothing to take down, madam. Nothing to take. Don't take down at all. That's not the way to become a good student. Just listen. Information is available everywhere.
Noting down the doubts.
No, doubts are doubt. Okay. This is just to introduce you to today's topic. So switch off. Let us start with the first line. I have left the University of Mysore. So, but affiliation, just 3 months ago, I was still here. I am now in Mahadana's PG Center. So, I was here for more than 10 years. I was a student here, as I already told you. This is something you are all familiar with. Evolution of life on Earth is a long story. How old is Earth? Can anybody tell me? How old is life on Earth? I'm sure some of you have been taught. I guess you are all bright students. 3 billion years? Yeah, no. Earth is about 4.5 billion years old, supposedly. And there was no life for more than a million years. The conditions of Earth were such that it could not make money. Or if you are busy, you can go, no problem. When the life began on Earth, it took billions of years to reach this stage where mammals and ultimately human beings evolved. Okay. So I am sure you all are familiar with the variety of plants, starting from the simplest of forms to the complex of product material. Similarly, in the animal kingdom, you have simplest of organisms like protozoans to the complex of mammals like apes and ultimately man. But this variety of life forms took billions of years to take shape. In the meanwhile, you have variety of shapes, forms, and functions. Like starting from a single cell to a multicellular organism to tremendous variety of forms you will find among invertebrates. If you actually see the evolution of life, you will see in the simple organisms where you have radially symmetrical body, then becomes bilateral symmetrical body. Ultimately, you will find a very complex mammal and variety of mammals, including man shapes. So the question is, evolution helps us understand how did all these forms evolve. I know that many aspects of evolution are not being taught these days. And if you take the evolution of complexity of life, this is what Darwin told us, that evolution of life is one. It took billions of years, resulted in various symmetrical structures that evolved, and that constituted a change in the form and function. So if you take microorganisms, you have single cell organisms, very simple life, and then you go to these multicellular organisms, the complex forms of life, where the evolution of function. What is the function of a living organism? The living organism has to eat food, it has to move, it has to make, it has to reproduce. That's how we describe living forms. So in order to live and reproduce, it has to eat, it has to mate, and the process of life on earth involves these activities that evolve into very complex forms of life. This is the story of evolution which Darwin told us. Right, thanks. Now the Darwin told us, or gave us the concept of evolution, that the evolution of life evolved from simple organism to complexities, and this complexity of form, structure forms, which evolved from the simple ancestors, and these ancestors ultimately gave us to a variety of forms that were successful adapting themselves to an environment, and therefore survived as a species, which gave rise to a variety of species. And this complex varieties of form actually tells us the story of life on Earth, which has taken billions of years. Just to give an example, if you take a fish that lives in water and a frog that has successfully colonized land, and reptiles, birds, and mammals, just to give an example. First thing you need is to move, locomotion. Fish in water does not need legs, but it needs fins. Whereas the frogs have to walk on land, therefore the evolution of legs happened. I am just giving you a complex story in a simple way. And this fin to leg, and then these legs further gave rise to, in the case of birds, the front legs became wings. So this evolution of just limbs of locomotion involved a part of the structure which formed fin, which also gave rise to front legs, back legs, and legs. That means the tetrapods, in other words, four-legged animals, evolved from pre-existing structures. A structure in an organism cannot suddenly arise. Suddenly legs cannot develop. It has to evolve from a pre-existing form. That is the fundamental in biology. You cannot suddenly be born. There has to be pre-existing parent first. The parent had a pre-existing ancestor. And the ancestor of species had an ancestor. And this ancestral species had a group of ancestors which gave us to several different kinds of organisms. That is the tree of life. Okay, you all know this basically. But the question is, the story of evolution of form, diversity of forms, was constructed based on anatomy, based on morphology. And the evolution of our ancestors from whom we inherited all our body form and functions were based on fossils. So if you take fossils of any organism, you will find a pre-existing ancestor which had similar form. Humans had ancestors who have become extinct. We ultimately evolved into present-day forms, but we are not the same. But there were our relatives who remained as apes, and one particular branch gave rise to humans. So the evolution of form was understanding based on a particular form. Form simply means a biological form, which has the body plan, which has the structure, and this structure evolved into an organism, and this organism is diversified into several different forms. That's what story of evolution tells us. Now, with the advent of our understanding of molecular biology, we all know from the days of Mendel and Morgan that every trait an organism possesses has a gene, as a genetic basis. If you are fair, if you have a long nose, if you have dark hair, if you have brown hair, you all have inherited the traits because your parents had those traits. And Mendel called the characters of the traits as genes. That was the way we all know the story. And it is Morgan who gave us the concept that there is something like a gene that is carried on a chromosome whose identity can be expressed in a trait, and therefore the concept of gene was fundamentally proven by experimental means by Morgan using Drosophila. That's how Drosophila is often called Cinderella of genetics, because we understood the heredity by Morgan's experiment in the 30s. And you all know this, the eye mutant, white color mutant that gave us that the color of the eye is carried on genes, and these genes are carried on chromosomes. The chromosomal basis of heredity gave way to the Mendelian genetic basis of heredity. So we understood for a long time. So therefore, every trait carried a genetic basis, that is a gene. On the other hand, the function, let's say, if there is a hand, it moves. It is a function. But the hand or any structure functions means physiology. It has muscle, it has bone, it has cells, and it has different types of cells. But all cells carry basic physiological functions. Every cell contains the genetic makeup. Every cell contains how the genes are expressed to make the gene products that are able to maintain the function of a structure. We are talking about structure of an organ, not structure of a cell. So the function of an organ is due to the physiology, which is due to the biochemical reactions. Biochemical reactions are pathways that are coded by the genes that specify what pathway in a cell is there. Okay, I am just giving you basic, only concerning students, I am not talking to experts. Okay, therefore, the fundamental concept of DNA, RNA, protein was established in the 50s. After the discovery of DNA structure, we were able to understand the DNA, RNA, protein is fundamental to all living organisms. But that does not tell us everything. This is universal. Muscle, you have the same genes, pancreas, you have the same genes, brain, you have the same, but the function is different. Physiology is the same, biochemistry is the same. How that is done? That developmental biology taught us that the genes in different organs are expressed differentially during development or even after they get fixed. This is what we understood. If there is a change in this mutation, there is a change in function. So people try to, it was proposed long ago that it is the mutation that brings about a change in gene. If Drosophila aicula can be changed by a genetic mutation, therefore it has changed a trait. an organism which has different traits become a different species. And a different species, therefore, is a product of genetic change that has happened over a period of time, evolution time scale. So people thought that a species evolves by genetic changes, and a series of genetic changes, what we call it mutations, are responsible for species, origin of a new species. Darwin had no idea how new species arose. All he said was species with modification that could successively survive, adapt itself, gave rise to an offspring which further diversified. Ultimately, that's why we have a variety of species, which is a successful species that has survived and multiplied. That's what the fundamental evolution is. So therefore, the genetic changes people believe is the raw material of evolution. This is also the theory in the 70s. What is evolution? Evolution is a change that occurs as a consequence of genetic changes that are fixed in the species, which are useful for the species, and the species survives because of the change. It is able to adapt, whether it's an environment or food or play. And therefore, the mutations are the raw materials for evolution. So, that is fine, that's what we have understood. Till 2000, we had no idea how a evolution structure can evolve. For example, as I told you, how a evolution of limb occurred from a fin to wings, because evolution of a structure. But if you look at the physiology and biochemistry, it is the same. If you take LDH of a fish, LDH means lactate dehydrogenase enzyme. If you take hemoglobin of a fish, frog or man, that's what initially people try to understand evolution by looking at protein sequence. That's all we had till the 70s. No genetic sequence was available till 1974. First gene was cloned in 1974. So when I was a research student, there was the concept of gene was only understanding them just then operon model had been discovered. So gene had a sequence which had, we used to call it cistron. So it had an upstream element that was understood mainly from the microbe. We never had the concept of promoter then. I'm talking about the early 70s. So the structure of a species that evolved over a period of time and diversified, I'm just giving an example, cannot be explained by change in function, because normally gene was thought gene is a protein. Gene, DNA, RNA, protein. Protein is coded by the gene that through an RNA gives such. That's all we know. Every protein we know that makes up the cells, every enzyme we know that is a part of a metabolic pathway, is a product of a gene. So our understanding for a long time, from 1950 to 2000, that was true. But we were not able to understand how an evolutionary development of a structure evolved. So the existing information on biology was based on the evolution of morphology or anatomy, was not able to tell us how exactly a structure evolved. Suddenly you cannot get a wing. So from reptiles, you have birds and mammals, reptiles have an adaptation to live on land. Reptiles can walk. Some reptiles like dinosaur age, they were also flying, give rise to wing. And mammals give rise to totally, but we are not able to explain what is the gene that gave rise to a mammal that could live as a mammal. We are not going to details of it, or as a bird. So therefore, we were not able to understand the evolution of structure function. All this changed when once the wonderful field of genomics opened up, when the Sanger discovered the technique of genetic sequencing in 1977, subsequently Human Genome Project. Then we had PCR, and these two powerful tools gave us opportunity to sequence the genomes of organisms. Earlier we were only sequencing the genes that was function, where we could get a sequence for a gene which would give rise to a protein. Now we have powerful tools which can give us the sequence of the whole genome. And you all know, genome, at least in eukaryotes, including humans, as much as 70% does not give rise to a protein or a gene product. It is not transcribed at all. And people call it junk DNA. For a long time, it was even taught. Junk DNA was taught when Bharat was a student. I'm sure when Madam was a student. Now we know it's no longer junk. Then what is it doing? Evolution doesn't keep something that is useless. And up to 60, 70, 80% of some of the genome is not contributing to any specific protein. So what is it doing? These people never understood. Now the scene has changed. Okay, as I told you, that specific function of genes that encode for specific proteins cannot explain evolution of morphology. As I told you, it does not explain, but it can only explain specific changes in the genes. Then, is there a role for non-coding genetic sequence? These people never had an answer to this. What is the so-called junk DNA doing? Is there a role for it? It was not known at all. Now let us come to the fundamental point. Once we have genetic sequence, we know how we are similar to apes, our closest ancestor. How Drosophila can have genes which has something as you say 70% homology to some genes. But the whole genome has got 90% homology to human. But we are so different. Not by getting which are different species. Evolutionarily separated by millions of years. We branched out from primates, and one of our ancestors, it's not an immediate ancestor, it's a as you all know. So the man today, if you compare the genetic sequence of a chimpanzee, who is our closest relative, has something like 90% homology. So what has changed? That means 90% has not changed. Is that 2% sufficient to explain who we are as humans? We don't know. So this is what is the exciting field today to understand using the powerful tools of genomics, molecular biology, sequencing, to understand what exactly is the basis of a species, how exactly it differs from it, how did it function about. So if you compare man and the chimpanzee, what is the fundamental difference? They are two different species, although very closely related. Man has a bigger brain. Man has got the ability to talk, communicate. Man has the ability to use fingers, tool use. Although the primitive forms of tool use was discovered by some social groups of monkeys. So the evolution of language, the evolution of brain seems to be fundamentally different. Can we understand by using the tools of genomics? So the fundamental questions that arise is how do we explain anatomical and behavioral differences? Genes that control anatomy and function. Are there genes that control anatomy and functions of the body? We all know that all physiological functions are controlled genes. We have identified metabolic pathways, and every enzyme has a gene behind it. Even the protein that is known is a gene behind it. But that does not help us to understand the anatomy at all. But it does help us in are there sharp distinction between evolution, anatomy, and physiology. Physiology didn't change so much. For example, your hemoglobin in a rat or a monkey or a man does the same thing. Your Krebs cycle is present in all these organisms. That has not changed. You agree? But what has changed is something for which we do not have a purely scientific, acceptable evidence as a, but people are now beginning to understand that. So there is a sharp distinction between evolution, anatomy, and physiology. You take hand of a monkey and the hand of a man, fundamental physiology is the same. But the shape of the bone and the shape of the body form is different. That is about. So what is the genetic? Because an existing form can only evolve from a pre-existing form. How exactly do you understand this in molecular terms? If there is a change, that has to reflect it in molecular terms. In other words, in sequence. Can we understand that? Change in morphology, that is size, shape and number and color, for example, fundamentally different from change in physiological process. Because this has remained more or less, although there is tissue-specific physiological evolution, but an organismic specific anatomy has changed, which we need to understand in terms of evolution. Let's just try to examine the relationship between, for example, people looked at in the early 70s anatomical variation in birds, mammals and apes, and they looked at genes. They found that it is the evolution of regulatory elements that have changed, but not the genes that are coded for. That means any particular change, as you saw, Morgan looked at a change in eye color, it was point mutation. If there is a point mutation, that is one genetic change in a particular gene that controls a particular protein. That's easy to understand. That's a change in the protein. But on the other hand, we understood that a gene can be present or absent in its activity, in its expression, if it is not switched on at all. Your insulin gene is present in your muscle, but it is not switched on. Why? Because the switch, that is the controlling elements, the just regulator elements, are not turning it on, it's switched off during differentiation. So, one of the things people look at, is it these regulatory elements, for example, evolution of a new organ, like a brain or like a pancreas, was possible only by switching on and off of genes, which is only controlling elements. That gave us an idea that there is a whole new organ in the world with specific function, which is not due to change in the genome, but in genome expression. Therefore, it gave us a clue that is it possible that regulatory elements have changed during the course of history of evolution of forms. That is what people look at. Over the last 40 years, this is what we have understood. Anatomical variation in multicellular organisms. This was proposed in 1975 before we knew anything about genomics. This was one great person called Alan Wilson. He proposed a theory that evolution of form has something to do with the changes in not just genes, but regulatory elements of genes. This, in fact, last year's Nobel Prize, that is Svante Pääbo, was a student in Alan Wilson's lab. So it's very interesting. And these were the labs, those of them, they started understanding genetic sequences. These are early days. Because 77 only the Sanger sequence came to be. Genetic sequence was not available to us. But he had the foresight to theorize, hypothesize that just based on molecular sequences of proteins. Those were the days when they had sequences for some of the proteins, like hemoglobin, like enzymes. Those genetic sequence and the biochemical sequencing of molecules, proteins, gave us an idea of molecular evolution. What is the change that has happened, for example, in an enzyme or a protein from fish to man or from reptile bird to man. So this actually gave us the changes in the molecule, which are to be the changes from the genes, for which there was no tool at the time. So from that he said, changes in these specific molecules will not let us understand the evolution of form. So he theorized that probably it is the regulatory, because the genome will not change. Genome changes. It is a variation that it gives rise. Suddenly it cannot change. But the expression of a gene or a set of genes can change the function of an organ and an organ. So this is what I am trying to just Okay. Anybody has seen this book? This book is one of the most thought-provoking book ever written. In 1970, his name is Ono. Evolution, Sushimi Ono, Evolution by Gene Duplication. He wrote a book in 1970 when there were no tools to understand genetic sequence at all. He wrote that book in 1970. He said, unless there is a genetic change throughout the course of evolution, new forms cannot evolve. So there is to be a drastic change in the evolution of genomic variation, which involves genes duplicate. Now gene duplication, he is an established phenomenon, but he was able to theorize. In fact, when I was a student, I saw this book in the library, I read it, I didn't understand anything. All we knew those days were chromosomes. And today, even now, is quoted as a person with a great insight. Evolution by gene duplication is the title of the book, and he said, allelic mutations in the gene basis cannot account for major changes in evolution. You all know what allele is. Mutation occurs because gene always is a pair of alleles. One mutation can lead to change in the gene function, but does not account for the changes in evolution, which is evolution of form and function of new species. So he said, duplication of regulatory elements must have contributed greatly to evolution of vertebrates. Those were the days when only operand model was known. Operand model, anybody, any idea? When was it published? Genetic students shared book. ಇರೋದಿದ್ರೆ ಚೇರ್ಮನ್ ಎಲ್ಲ ಹೊರಗೆ ಸೇರ್ತಿರ್ತೀವಿ. Operand model ಯಾವಾಗ ಕಂಡುಹಿಡಿದ್ರು? I'm sure you all have studied in PSC. Jacob Bonard, he got Nobel Prize. First time it was demonstrated in bacteria that the gene expression involves an upstream element, right? And this expression of the gene is controlled by promoter sequence. This was first discovered in microscope. It's a very complex process in eukaryote. Anyway, that doesn't matter. You know it probably. So the duplication of regulatory genes must have contributed greatly to evolution. This is what I don't know how could you do that, because at that time all we knew was only Jacob Bonard's work, and we had no idea if there are upstream elements in eukaryotes and how exactly they function. But we believe that it was there. Gene expression is always there. Okay. Now today, this is the theoretical framework. If the evolution of gene changes during duplication, how exactly it happens? Let's say there is an ancestral gene. If it's duplicated, it becomes the same two copies. And this sequence changes in the duplicated gene. You can have a gene that has changed, sequence. It's the same gene. It has duplicated. It has changed sequence. And then further change leads to specialization. You see, which was the single gene. Now it is given as to 1, 2, 3, 4 types of genes. This was only theoretical possibility. And this evolution of genes by duplication was possible by duplication always occurs. You know why it occurs? It's actually an error in replication. And many times this duplication occurs, either it is functional or non-functional. It can occur in genomic sequence or it occur in a transcribable sequence or non-transcribable. That is, it can happen in an exon and intron. That we all know. And this variation is possible by gene duplication and further variation of the sequence. And if that is useful, it could be conserved and it can be specialized due to functional change. Ultimately, you have a new function from whatever concentrate. This would take millions of years. Depending on the species, one species gives to many species, and when it gives to many, there are thousands of generations, and thousands of generations changes went on happening. What was useful was retained, what was harmful was eliminated by natural selection, what was neutral also was retained, useful was adaptable, therefore species increased which carried this particular useful mutation. This is how we understand the tools of molecular biology, how exactly genes ever work. This was a theoretical problem, but do we have evidence? Another, that I talked about sequences of genes that are changed, genes that are transcribed. But here, let us look at the sequences of gene evolution in the upstream elements. These are promoters, you all know. This is a gene, there is an upstream element. Upstream element is a promoter sequence. This promoter sequence can have a mutation. And this gene, if there is a mutation, may not be working at all. Or if the mutation carries a change that is responsible for gene expression, whether useful or not, the evolution ultimately will determine it will be selected or not selected. And this particular sequence can further diverge. Let's say situation B, regulatory element itself can duplicate. Any sequence of gene can be duplicated during duplication. And this duplication can work from one upstream sequence can end up 3 times. And there could be change in these 2 upstream elements, the sequences. And therefore the expression of this gene here will be affected differently. Similar, these are all possibilities. I am going to give you that evidences have been forthcoming in the last 10 years or so. So here there is another thing. This is the transcribable gene. And there it's all exons. Now you get a intron. You know intron exon, right? And if you have 2 genes, they produce the same function. We know that it is spliced and forms the protein that it is coding for. But you will have end up 2 kinds of genes. We call it isoform evolution. Same gene, but it has different regulation that is expressed in. So one sequence can give rise to either one protein or many proteins. Many proteins may have many different functions. So gene duplication followed by mutation, expansion of cis-regulated elements, and evolution of new exons. So you have here an example, simple example how a new exon can evolve. So, as it happened in the evolution of anatomy, do we have evidences? That's what the powerful tools of genomics has given us to understand. History of Hox genes, for example, you all know, we don't have time to take it. You all know in development, Hox genes were discovered in Drosophila, you know that. Actinopedia, Bithorax. That means the body plan has got elements that can decide whether a particular body structure is present here or not. For example, antenna is supposed to be present here, not here, in Drosophila. But if there is a mistake, antenna can be here. So antenna can be in a place where other than they are supposed to be there. So also legs. Instead of 3 pairs, you can have more. You can have legs in elsewhere. This gives us an evidence that the form, that is the structure, can be changed due to a mutation, due to a change in the genomic expression, which is possible in cis-regulated elements. This you don't worry about this. This is a gene for melanin that will get various types of pigmentation. For example, all of us, different human races have different coloration, which is the gene that is present. Don't worry about that part. Okay, so gene duplication has contributed to evolution of form over a long period of evolutionary time, but does not account for diversification of lineages. That means still it is not enough. We have something like 300 species of beetles. We do not have sufficient knowledge to understand how exactly 300 species of beetles can evolve. So also there are 10,000 species of birds. How exactly 10,000 species of birds can have variations that were evolved over a period of time by the genetic sequences? Because ultimately, a bird that evolves from an ancestral bird carries the same sequence, but a chain sequence that gives us a new species. How do we understand this? So this gene duplication contributed, but not sufficient to account for diversification of lineages. This is what our understanding today is. Therefore, regulatory sequence evolution form, we have now evidence from diverse animals and plants where association between gain or loss of modification of morphological traits. As I told you, you can have a loss of a function, or in the case of fruit flies, there are very good examples also in plants and vertebrates limb and axial diversity. Studies have highlighted that these are the examples that shows it is not the coding sequences that is the cause of change, but it is a change in the regulatory limits. Let's just give an example. This is what we have got from the data on genomics. You see, you have a Drosophila here. This is a yellow pigmentation, and it also has a pigmentation, a black spot in the. And this, if you compare the genomic sequences, because it can be manipulated in the lab, and they have found that there is coloration in the wing body, and here the wing spot body, and here an extra. This was due to a mutation and duplication in the 6 regulatory. That explains the morphology. This is the difference. When we sequence the whole genome of this species and this species, Melanogaster, this is not a good. This is a different species. Where this species has got this spot. This is species-specific morphology in the beings. And we identify this as a new species. And this new species with a new structure is possible by the genetic change that has happened. Otherwise, everything is same. This was not known to us just 10 years ago. And I'll give you another example. This is a fish. This fish has got, fins, the forelimbs and the posterior limbs, anterior and posterior fins. And these are two different species of fish. And if you compare the genomic sequences, they are identical except in the absence of the posterior fins. This particular gene has mutated. So this is not found that this is actual evolution of body plan. It is not an exon. It's a species, just sample. It is evolution of function anatomy, where regulatory elements has caused the absence of why a fin is a structure developed by a regulatory gene that completely directs the formation of a limb. Like I told you, oxygen, it's equivalent. So this, if that has mutated, the whole limb is not formed. Therefore, we have two different species, one with 4 fins, the other with only 2 fins, and this has disappeared simply. And we now have able to explain the evolution of a structure that has changed, which leads to evolution of a species. They could be different in different different places, where they have survived and succeeded because nature has selected that it can still survive without approaching it. Ultimately, the story of selection, that the species ultimately stays, evolves, and if it is no longer useful, it will disappear. We have learned from fossil. But this actually tells us the truth about the and fish story, that in fact the new species has evolved here by a change in the structure or a form. This was not known to us just 10 years ago. So therefore, this is another example where, in plants, they have actually found that the gene can change and ultimately give rise to resistance, and then it can diverge, and in rice plants they have found similar kinds of things. Plants are also, we need not discuss this further. But I will tell you this particular example. You all know, famous case of orange finches. A species that was confined to the islands of Galapagos led to radiation by adaptation to different species depending on the food that was available. That is Darwin's, the people say that he got different beak which can allow it to eat different kinds of food, was able to survive and select it. And recently, there is a couple of husband and wife zoologists who have been studying these species for the last 50 years. And in one particular island, due to the floods, the birds disturbed except one particular species. Ultimately, they found over periods of generations, the species diversified and adapted to new food that was available. Some plant that was available disappeared. And few which had beaks that could eat the other kind of corn or some other, and it survived and multiplied numbers. Then there was a change. So evolutionary change is made possible by environmental or climatic change, where a successful species which has adapted, in this case a pig, survives and multiplies. That is able to explain us different species. And for this, why I am saying this, for this now we have molecular evidence. Here you can see genes that control will shape. You can see, out intestine, it is the same gene. Now we have this is very recent. So mutations in the upstream elements cause variation if it is adapted lead to a new structure and therefore new species over a period of time. Give you another example. These are birds. You know, there are birds which sing, there are birds which do not sing. Okay, we were talking about songbirds. We are not talking about birds. Every bird makes a noise. Now, the song, the bird that sings, for example, koel sings, right? Whereas the crow does not sing, but it calls. The song is an interesting function of the brain. If a song has to come, there is a brain function, there is a larynx, and there is a muscle that controls the movement. And that leads to an extended sound in a symmetrical form, what we call as bird sound. That is human speech, for example. So, a simple bird call is essential for its survival, but it evolved into a call in certain species which was useful to that particular species in that particular environment, and it was retained because the species became successful, and this song was essential for it to announce its presence or to attract a mate in a given area. So the bird call is considered a language of the bird. There are birds which sing, birds do not sing. Here is a very interesting case of evolution. You see, this is a finch, this is another chickadee, this is a ringtone. These are songbirds. This is not a songbird. People try to look for a specific gene function, expression that occurs in birds that are singing. And there are birds that sing only during breeding season to attract a mate, right? And during non-breeding season they don't sing because there is no need to sing if it is to attract a mate. And this ability to sing only during breeding season must have something to do with it. Hormones, because hormones express during breeding season. In fact, science has given us evidence that in songbirds only during breeding season in July, that gene gets expressed. This is a non-singing birds, a singing bird, and the singing birds have the gene expression, and the non-singing bird does not have gene that is expressed. Here is the case, in a singing bird, only during breeding season, the gene is expressed. So we have no evidence. The expression of a specific gene in the brain is associated with the call, which means evolution of a bird call. Evolution of a call is now ultimately as a genetic basis. I told you the example of morphology. Now I am giving you the example of function, in this case a call. So, the contribution of, I just described this. So the conclusion is, empirical studies on evolution of traits and gene regulation, rate of gene duplication, regulatory proteins are mostly slowly evolving. What they say is that regulatory elements and gene duplication can take several thousand generations, which could be millions of years of evolution. But although it is an important factor in the evolution of form and function, but is it going to tell us the complexity of life? We still do not have enough information. But there is adequate basis to conclude that evolution of anatomy occurs primarily through changes in regulatory sequences. This is what we now understand. Earlier, we never knew that regulatory elements have any role at all in the evolution of species or form and function. This is what has given us in the last 20 years. Now let's come back to the chimpanzee and human. As I told you, if you look at the genomic sequence homology, it's something like 80%, even 98%. Is that 2%, is it enough to tell who we are as humans? Can we distinguish? Is it enough? Or what actually lies behind? So the product of evolutionary change is the output of a complex trait, in this case, humans or primates. It's brain size, craniofacial morphology, cortical speech, languages areas, and a digit form. This is what describes primates different from other organisms, and humans as distinct are about more form. And difficult challenge is to map changes in these two traits. One of the evidence we have is that intriguing example of Fox gene and evolution of speech. The evolution of speech is a very characteristic event in the evolution of humans. Speech and the evolution of speech is integrated to the evolution of the brain, the cortex, and the evolution of direction and the control centers in the brain and the muscles. And that's what makes us humans. So let's see what we know now. That I gave you the example of the evolution of speech right now. There is a new area that has emerged in the last few years. Can a new gene evolve? According to the rules of biology, genetics, you cannot get something new without pre-existing form. Do you know origin of anything is impossible? If something is already there, it can give us something, maybe a modified form, but it cannot suddenly appear as new. So the evolution for the structure function has to be from pre-existing structure and function, or a pre-existing species. That's the fundamental basis of biology and evolution, right? So here, now we have understood that a new gene can evolve suddenly. I'll give you a very interesting story. You know about the Arctic ocean. It freezes when below 0 degrees. No organism can survive. But there is a fish that can survive in the Arctic waters. This Arctic cod, people try to understand, physiologists, how can it survive? Because if you take a fish from some area, it will die, the temperature, it will freeze. So it remains from freezing by an interesting protein called antifreeze protein. This is an adaptation. This particular species, by accident, has evolved a protein that is able to prevent it from freezing during the winters, sub-zero levels. This antifreeze protein, people ultimately with the tools of molecular biology gave us the sequences of the gene that causes the protein. They found that genomics tells us if you take the whole genomic sequence, it's completely new. There is no pre-existing gene for an antifreeze protein. How do you discover a new gene? Compare the genomes of the relatives. Let's say if there is a gene from one fish or Drosophila, look at the other species, because a gene has to have a counterpart in the other species, but it can get modified. There has to be a precursor or an ancestor for this gene. They found that there is no sequence similarity whatsoever. Now, powerful tools of genomics, we can sequence the whole genome in a matter of a couple of days, or maybe a couple of hours in a few days. No, it is whole sequence variation. It is not Kaplan-Meier. It's a new gene as evolved. This sequence for this antifreeze protein does not exist in any fish. There was a lady who discovered this. She was not able to explain. They looked at sequences of closer relatives. There was no sequence. That means there is a new sequence. How can it cause it? Because they were looking at sequences of proteins. Then, when she looked at it, found that there are sequences in the non-coding sequences, not in the circular elements, which were found in the antifreeze protein sequence. In other words, you have a new gene that codes for a new protein, and these sequences are derived from a non-coding sequence, so-called junk DNA. This was a very revealing discovery. They found ultimately that it is a non-coding sequences in the genome that by chance came together to give us a functional gene and protein. What's a functional gene? We should have open unit frame, it should have regulatory element, it should have termination sequence, and you get the gene. This is how today we understand the gene. Whereas in a non-coding sequence, you don't have an open unit frame. You don't have a stop signal or end signal. You don't have it. So the structure of a gene is functional gene, has to have this basic. And here she found that there were sequences derived, this is how, and ultimately gave us a protogene. This is the basic possible evolutionary form. And this protogene has given us a new protein. This new protein can be useful are useless or even harmful. In the case of fish, antifreeze protein was very useful. It helped the species to survive. This has happened millions of years ago. Whereas it could also, because this is a random phenomenon, these new genes can continue to get formed. You will get the transcribed sequence, and this can become what you call protogene. And if it occurs as sequences, it can form a protein. And this protein can be useless. It could be discarded. It's not selected, or if it's neutered, it may still be there. And if there is a selection, this protein has a new function. New function, therefore, a new protein, a new gene. So, new evolution of these are called de novo genes. This is a fantastic which has happened in the last 10 years because of the advances in genomics. Now we will come to the last slide. As I told you, how humans are different from chimps, for example, to give an example. Two species, closely related. But the fundamental difference between these two species is the brain size, brain function. So though the primates, and humans in particular, primates in general, have big brains compared to the bodies, relative size of the brain. That is what primates are known for. That's how intelligence evolved. And the cortex has multiple functions, expanded cortex. And this brain evolution, this is a field in itself. Whereas if you take smaller animals, like rats or monkeys, a smaller brain, people did a very interesting, so we need to find evidence how exactly big brain evolved suddenly. So the size of the brain evolution is one thing people were interested in. Found that if they found a sequence, if you put into a small brain animal, they do what is called organoids. Organoid means you can take a stem cell and a pluripotent cell, you can develop into an organoid. It doesn't completely become an organ. This is used in the research today. So they took a neural cell line, they allowed it to differentiate, and they transplanted or they put a gene which what they thought could probably be responsible to create a large brain. And they found that this organoid developed into bigger organoid, that means the size was bigger. So there is an evidence, this is purely experimental evidence. This particular sequence, which was synthetic, derived from the sequence available from the humans, and then it is expressed, and it led to the change of the structure, gave a bigger organoid, which means to say that we can actually understand the evolution of form and function, including the size of the brain, which has a molecular basis, for which we are not used. Today that is made, but this was only published very recently. Very exciting, many people are working on this. Therefore, into the grand old concept, nothing in biology makes sense except in the light of evolution. That was also said in this famous quote. Everything makes evolution sense. We know if you know how to look at it. Now there is evidence to say that evolution makes something from nothing. This lady discovered that evolution has invented a new gene. At first it was nothing DNA, junk DNA. Now it has made something out of it. In fact, some people use a jargon called genes from junkyard. In other words, it has created a gene from junk DNA. So this is what I have added. This nobody said it. This is what is a standard slogan in biology. Okay, thank you. This is all I wanted to share with you. Any questions?
Feel free to interact. This is for your sake.
Doesn't matter any question, because there are no silly questions. They can only be silly answers.
Anything, no problem. Okay.
Doesn't matter.
At least the sir will come to know.
At least
you will.
this is something which I read and I am talking to you. Because science is going so fast. Even reading and digesting is not easy if it's outside the field. It's not my field at all. So I thought for genomics students I should talk about genomics. The best way to learn is to teach. So, yes.
I'm Vijay from First Year LC. So I have a question. So now we are having a different challenges in environment and in other cases also. Can we expect any evolutionary changes in humans in future?
Good question. Sure. The question is, if I understand correctly, there is a lot of change in the environment that affects humans. Can you expect change in humans in response to the environment?
Yes,
Exactly.
yes,
Correct?
yes.
Yes. But we don't call it, there is a response. Any agent, physical or chemical, that affects your genetic makeup makes you either suffer from a disease or adapt to a new change. If the environment, see, environment is a very broad term. So environment as you define, today let's say there is less food, you are not able to survive. People have survived with less food by changing their body size. This we have the examples. If you take the hunter-gatherer cultures, you have in Andamans, we have a species, it is humans only, whose body size is very small, they are just about 3.5 feet. So that is one side, food. If you take, let's say, toxic substances that will exist everywhere, that toxic substances either can kill you or cause disease or dysfunction. But if your body is able to detoxify that substance, that detoxification is a biochemical phenomenon. And that detoxification, if it is known in insect, for example, insects' resistance to insecticide is by its ability to detoxify a chemical which kills it, which over generations, that genetic change which is due to an enzyme that can degrade it many times more, it led to resistance in this particular species, which is a genetic change. In case of humans, you need many generations if a mutation is selected for adoption and survives in generations. So, any evolutionary genetic change to be selected is a function of generations. Bacteria can evolve a new mutation in hours, days. Insects in weeks and months, because generation time is very small. Similarly for humans, it took us many generations. So therefore, theoretically, humans after all is just an organism. We have the ability to adapt. That is biological adaptation. We have the ability to think and do something about what is causing us harm. That's a different issue altogether. Okay, biological species, any organism can deal with environment. Either it is perished or it's able to survive by adapting. What is this adaptation? Is it a change in a molecule? Is it a change in an enzyme? Is it change in color? That is adaptation. It's able to survive, and that is selected in next generation, and that particular change increases. This is what the story of evolution, adaptation is all about.
Sir, what's the role of epigenetics in evolution? Because all these things are regulatory elements,
Correct.
duplication.
Epigenetics is one field where genome does not change, gene expression changes. This is one level of gene regulation. Epigenetics is the modification of a particular gene or a controlling element that leads to either effect on its expression or not. Epigenetics can be also affected by environment. Genetics can also be environmental by physiological environment. A gene, for example, methylation of DNA, is very well-known epigenetic gene. It can switch it off. That happens in developmental regulation. So why exactly it happens is that if methylation happens in the DNA sequence in upstream end, your transcription factor will not bind. Your transcription, if it will not bind, even if it binds, it's not effective, and therefore expression of gene doesn't happen. This is how gene expression can be affected, turned on or turned off due to change in epigenetic effects. There is a DNA methylation in such example. In fact, now they have discovered epigenetic change in the histones. You know, gene expression involves histones of the gene, and the histone modifications of protein. Can also affect in making space for transcription. How exact transcription? It's the histone proteins that have to uncover the DNA sequence. And if they modify that particular protein, now we have evidence. So the epigenetic changes are the changes extraneous to the genomic sequence that can happen either in the sequence by modification or by the protein that is a part of chromatin. So that is epigenetic. Epigenetic change is involved in disease, pathology, health. So gene expression is the basis. Epigenetic is one of the means in which gene regulation is controlled.
There are questions.
Dr.Mahadeshwara Prasad is no question.
I'm relaxed.
If there are any more questions, you are free to ask.
I want to add up for such.
Yeah, please.
So actually I'm a forensic medicine man. So for your question, research is going on. I think when Sir entered as professor to this institution, he used to see the student of well-built, no sir, the height, the weight. Nowadays you people look like Lilliputs for us. That is because from the past decade, you are addicted to mobile. You always sit at a corner of a sofa or a cot, completely flexing your lower limbs and upper limbs and the body, so that the body is shrunken. There is no action for the muscles. The muscles are not putting into action, they are not pulling the bones apart. So the epiphyseal is fusing early. This is one of the features that has been proved that evolution doesn't want hundreds of thousands of years. A decade is enough to change the shape of the body. The genes are not stimulated. The body is not asking the gene to accompany or to flourish whatever the height or the weight is required. And the food, what we are eating, the gene exposed to this one. So that's why earlier menarche for children, especially girls, used to be around 12 to 14 years in her head. But nowadays 80 to 85% of the girls are maturing at about 8 to 10 years. This is also because of the evolution. Sir told about hormones. This is one of the most important things, that amount of hormonal secretion is increasing, especially the sexual hormones. That's why making them more vulnerable for the maturity in the form of sexual things. And at the same time, the thinking ability. You know what you are going to think. Do you think about biology, zoology, chemistry, physics? Do you think about, I don't know what to say over here. So in forensic psychology, what we say is nowadays, if 2 people sit or 5 people sit together in the campus, everybody will be bending like this, and after 1 week they come to us telling that, sir, severe pain in the neck, sir, which is not at all controlled by anything else. So this is undergoing cervical radiculopathy or cervical spondylitis. Don't keep on bending, you will always bend like this in life. So this is once again the length of the neck is also becoming short nowadays. You just see the neck.
That's pathology of individuals, but they give rise to healthy individuals, which they also repeat parents mistake. So it looks as though it is evolution.
So this is what change in lifestyle is helping, or whatever it is doing to the body in the form of evolution, the whole thing is changing.
Oh, you have a question? Yeah, sure.
Sir, what is the role of metagenomics in evolution? Because the
Role of?
metagenomics, the genome of organisms that were within us.
See, this is meta actually is a term used when you have many sets of genomes. You compare. Okay, comparative genomics. So meta is a word that is used. Metagenome is not anything special. So you compare the genomes, try to see if there is a change which can ascribe to any specific change of the organism, function, disease, or variation. That is metagenomics. It's actually comparative genomics. So in the case of evolutionary biology, we compare the genomes of species, looking for changes in the genomic changes that are attributable to a change which is species specific. Okay, that is the value of metagenomics. It's nothing but comparative genomics. And meta-analysis, it's a very general term. It's not a biological term as such.
Madam, basically, metagenomics, when you are going to take multiple genomes and there are subsets, you will not be able to segregate. For example, basically the word metagenomics started with the microbial colony. When we are going to take culture, or for example from your mobile phone itself, I was telling how many microorganisms will be there, right? Or your towel that you are going to use if you are not going to sterilize. So the other day in BBC he was showing that the day-to-day, the towel that we are going to use in our home, if you are not going to keep it clean, that is sterilized. He removed a sample from the towel and he showed that nearly around 300 varieties of microorganisms are there. So how will you be able to segregate those genomes? You will not be able to separate it, right? So multiple genomes are there. So that when you are going to do the analysis, you will not be able to say that this is this strain genome, this is this strain genome. The word was originally coined for microorganisms culture, where separation of the genome cannot be done. So that is called as the genome meta. Right? Now what evolutionary biologists they are doing is, they are collecting multiple species, multiple organism genome from an environmental site they are collecting. That is also, in some of the papers, they are also calling it as metagenomic data. Right? So, now when we are going to do the Jiva study, or when we are going to say that an environmental factor is responsible for a change, means, for example, if something happens in this environment, means human alone will not be affected. The modification should happen to all the organisms that are living in this entity, no? Whatever effect on our genome is going to happen, for example, a temperature or pollution or carbon dioxide level, whatever change in environment happens, it has to happen on all organisms that are inhibiting this niche, right? So specifically, metagenomics, now evolutionary biologists, they are also applying to the cross-section of say biodiversity that is present in the geographical location. So they say that if a change has to happen for one species, means it has to happen for all the other species also, they will be affected and their genome will be modified parallel. So that now they are applying even to multi-species level genome, they are applying the word metagenomics.
Thank you, ma'am. I thank Dr.T.Shivanandappa for delivering such a knowledgeable talk and enlightening us with a lot of information. I'm sure all of us have learned something new today. Thank you for spending your valuable time with us, sir. I request our chairperson to felicitate him.
ಅದೆಲ್ಲ ಯಾಕೆ ಮೇಡಮ್? ನಮ್ಮ ಮನೆಗೆ ಬಂದು ನಮಗೆ ಏನು ಸನ್ಮಾನ ಯಾಕೆ ಕೊಡಲ್ಲ? ಇದು ಒಂಥರ ಮುಜುಗರ ಆಗುತ್ತೆ. ಅಯ್ಯೋ,
Every moment we should cherish, sir.
ಅಲ್ಲ ಓಕೆ, ಇದೆಲ್ಲ ನಮಗೆ ಸ್ವಲ್ಪ ಕಷ್ಟ. ಥ್ಯಾಂಕ್ ಯು.
Thank you, everyone. There's a short refreshment break for 15 minutes and let's all join for the lesson number 2 after that. Thank you.
ಕ್ಲಾಸಿಗೆ ಬರಬೇಕು.
All of you, please. 10 minutes break will be there. Then we'll commence next session.
In the grand tapestry of existence, it's often the smallest particles that weave the most profound stories. Ladies and gentlemen, distinguished guests, and my dear friends, a pleasant morning to one and all present here. I, Dr.D.R.Mahadeshwara, on behalf of the Department of Genetics and Genomics, University of Mysore, take this honor and great privilege to stand before you today as we embark on a journey into the realm of cutting-edge science and its implications for our well-being. We have the distinct pleasure of welcoming a true luminary in the field, someone whose expertise in nanotoxicology is not only vast, but also pivotal in understanding the consequences of advancements in nanotechnology. In a world where innovation continually shapes our future, the exploration of nanomaterials has become integral to progress. Today, we are graced with the presence of a resource person whose insights promise to unravel the complexities of nanotoxicology, offering us a glimpse into the potential impact of these minuscule structures on human health and environment. Ladies and gentlemen, it's my distinct pleasure to introduce our distinguished spokesperson today, a COVID-19 warrior, Dr.D.R.Mahadeshwara Prasad. With a robust academic background in science, Dr.D.R.Mahadeshwara Prasad holds the qualification of MBBS from JSS Medical College, Mysore, PGDHCM from JSS Institute of Management Studies, SJC Campus, Mysore, with a first grade performance and a distinction. Further, he acquires the qualification of an MD in the JN Medical College, Belgaum. Not only this, his qualifications kept on adding as he completes his PhD in genetics, genotoxicology from our very own campus, the University of Mysore. He was awarded for his research in cellular and genotoxicity of diesel combustion nanoparticles in humans. His journey in the realm of genotoxicology and forensic medicine and technology has been marked by notable achievements and recognition for his 37 publications as multiple Best Paper Awards, Best Indian Researcher, Scholarship and Sponsorship Awards, and many more. His contributions to the society are multifarious. Our esteemed speaker, Dr.D.R.Mahadeshwara Prasad, brings with him a wealth of knowledge and experience to our discussion today, which is cultivated through years of dedicated research and scholarly pursuits. His work stands at the intersection of science and responsibility, addressing the fundamental questions that arise as we venture into the nanotechnological frontier. Moreover, his commitment to advancing knowledge in the field of genetics is evident through his sublime and exceptional profile with innumerable projects, designations, achievements, and qualifications. Over the next moments, Dr.D.R.Mahadeshwara Prasad will guide us through the intricate landscape of nanotoxicology, unraveling the potential risks associated with nanomaterials and providing us with a compass to navigate the ethical considerations that accompany such advancements. The universe unfolds in the delicate dance of particles unseen. To ignore the smallest is to miss the grandeur of it all. As we eagerly anticipate his insights, let us be reminded of the importance of this dialogue in shaping the trajectory of nanotechnology and its applications in our lives. Let us acknowledge the depth of expertise and wealth of experience that Dr.D.R.Mahadeshwara Prasad brings to our forum today, who will undoubtedly enrich our understanding of nanotechnology and its profound implications for the world that we inhabit. I hope we all have an edifying experience ahead. Ladies and gentlemen, please join me in welcoming a COVID-19 warrior, Dr.D.R.Mahadeshwara Prasad, a true authority in genetics, as he will share his invaluable perspective with us.
Thank you for that introduction, but definitely I'm not the authority in genetics. Okay. And I'm really proud to be here, and because of ma'am over here, we had a wonderful time in academics, and I was doing the PhD over here under her able guidance. So wonderful teacher and a wonderful guru. We can just tell it in our own traditional language, guru. And today I'm here there for the opportunity. I thank Dr. Marley, the ma'am of this. And since the topic is overview of nanotoxicology, if we just carry on with this nanotoxicology, some people might be having their own doubt, because this audience has to first know about what is nanosciences rather than nanotoxicology. So when we want to discuss about these nanotoxicology, nanotechnology, these are all nothing but a group of things which contribute for, once again, a science. So we all join this prefix nano for all the things we can do nowadays. It may be anything which represents small or tiny or whatever. So when we want to know about the speciality or the endeavorness, as she told, regarding this nanoparticles or nanomaterials or nanoproducts, we have to go to the depth. All these days we used to talk about micro things, like microbes, microorganisms, micro things, microchips, all these things. But now we have gone to that stage that we are talking in terms of nano. Well, what is this nano? Did scientists created it? No. It's the nature. It's the nature which is created, and the man is yet to discover what is it in the nature. So this nanoscience is not a chemistry, not a physics, not a biology, not zoology, botany, or even not medicine. Then what is it? When it is nothing else, why pretty fellow you are standing over here? Tell me. But it is all. It's all. It involves the chemistry, it involves the physics, it involves the material sciences, it involves biology, it involves medicines. So, and etc. and etc. Measuring the nanomaterials itself is nano-analytical science. It is the super specialty branch nowadays. And I am here just to enlighten you people that what will be the future course of nano sciences. So where you can go, where you can take your career. That's what madam's view, that how you have to proceed in this science after completion. So keeping that in mind, I have covered the slides over here. So nanosciences, if you want to say that, it is about a phenomenon that occurs in the system with nanometer dimension. Any particle for that matter. If it is very small. For example, if I just take a bunch of chilies and throw on your face, you are not bothered. Isn't it? Mensikai, ela Kannada tane. Mensikai take mugud mele striyar tale keduskuntaru. Inganbiಟ್ಟು ಹೋಗ. At the same time, if I crush that and take the powder, chili powder, and throw on your face, what happens? Why this change is there? Because of the changes in the physics, change in the chemistry, change in the biology, how the body reacts to that particular powder of that chili. What will be the reaction which occurs when it comes to in contact with the skin or conjunctiva or nasal mucosa or oral cavity? And the next day morning also, when you are passing the stools, how do you feel if you have a nice spicy dinner? That is the level of irritation you can feel from external world to the inner aspect of the body. Each and every cell can sense this only because of the change in the physical character of that by a solid chili converted into a powder. Isn't it? So that's what we want to say, that how the particle acts in the nanometer dimension. So if it becomes still tiny, what happens, we have to think about it. So this nanoscience, as defined by the Royal Society, is the study of phenomena and manipulations of materials at atomic, molecular and macromolecular scales where the properties differ significantly from those at a larger scale. That's what I told you. When it is in a large scale or a bulk, what are the properties? When it becomes very tiny particle, what are the changes? So these changes, why these changes occurs, how these changes occurs, what is the importance of these changes, how the mankind can acquire these changes to his growth or disaster. Both the things is the nanoscience. That is, it is nothing but, this is the micro world, the scene, and we used to discuss all these days, and this is the atomic world, and the gap between the atomic world and the micro world is bridged by this nano world. So nanometer, you already know that it is 10 to the power of minus 9. So this is what? This is 10 to the power of minus 6, and this is almost, if we take the size of a hydrogen atom, it is 1,000th of a nanometer. 1,000th of a nanometer. So the gap between the atomic world and the micro world is bridged by this nanosciences. That is nanoscience. So these nanoscience, when you want to know about these things, you have to go through these various things. Introduction to terminology, history of nanotechnology, methods of nanoparticle production, approach of nanoparticle production, green synthesis. Nowadays everything is going for green because of the reduction in carbon. We have got the target to reduce the carbon particle to, I don't know that exact word, even recently we had G20 summit in India. Our Honorable Prime Minister Modi presided that conference, G20. Now it has been extended to G30. That is to reduce the carbon density, carbon load in the environment, because it is creating more and more health hazards. It's becoming a public health hazard in the current world. So this we might have read in the previous thing that what is happening in Delhi, and now also in Diwali. They are telling that why this is happening, because of this nanoparticles getting completely dispersed in the environment, and we animals, including that human beings as well as animals, are inhaling it. They are coming in contact with these particles, and hazards are increasing. And the complications to such a thing that we are facing so many dreadful diseases which cannot be cured. Only thing is which can be controlled. Earlier, people used to suffer with the infections. So, gastroenteritis, typhoid, a viral infection, like that, all these things. But apart from this, we have exposing to these particles nowadays, the health hazards are increasing. It is adding on to the burden of the crippling society. This is what is happening. So, this powder, as I already told you, if we throw on the body, if it is okay, talcum powder also everybody want to use on their body, but if it goes to the eye, what happens? If somebody inhales that powder when you are putting on your face, axilla or chest, what happens to that? Immediately the person starts sneezing. So this is what? Because this tiny particle has got its own separate thing. We can fight with the people who are big enough, isn't it? But we can't fight with 10 to the power of minus 9 size. Even we can fight with this. It's quite difficult because it will go into all sorts of orifices which it can enter and create problems over there. So this is one thing. But see, these 10 to the power of minus 9 particles will enter through the skin. It can enter through the nasal cavity through respiration. It can go through the oral cavity. So it can enter in through the eyes. All these problems. And nowadays, it has been anthrax. I don't know whether you people have heard about 15-20 years back. They used to post a small packet, and when you open it, it is nothing but anthrax powder. This was bioterrorism. That was the first time bioterrorism started in India also. So this is because of the powder of anthrax and other small materials. So nowadays, it is leading to biowar. I don't know, still Netanyahu is waiting for this one. If you would have done this to Palestine, it could have been finished, I think. But the only disadvantage is it can spread to the other whole world of that. Even today, there is a book by name Overview of Nanosciences only, written by Nandi. So he tells that we still inhale the nitrogen which has been left by Caesar. So because no particle can be destroyed, that's what he means. So he takes the Avogadro's number, and these amount of molecules are present in 1 mole of whatever matter, and the amount of the air which Caesar exhaled during the time of death is this much. So this has been distributed all over the world. So each and every person, apart from the plants, is inhaling that type of nitrogen. This is what he quotes a single barrel. No matter can be destroyed and created in this earth. So on this principle, he still says that. So that is the main thing, we can't use this bioterrorism. So when we are using these particles in the environment, what will be the tremendous effect? This is all because of the nano-sized particles. Okay? So that is the importance of this nanoparticles. So this nano systems arises solely from the tiny size of the systems. Nano, you know that, it's a Greek word, means dwarf. The opposite is giga. That is giant. So this is a dwarf thing, and the word nano is used to refer 10 to the power of -9 or 1 billionth of a meter. The diameter of hydrogen ion, as I already told you, is 1000 times 1000th of a nanometer. So in this scale, if this is all that there was to nanoscience, it would still be remarkable because of the incredible difference in the scale between the nano world and the regular microscopic world around us. So this is the difference between the regular thing what we are dealing with and now what we are going to this next nano world. If we throw the light on the history, in 1959, Richard Feynman gave a talk to the American Physical Society in which he laid out some of the consequences of measuring and manipulating materials at the nanoscale. This talk was known as a famous that there is plenty of room at the bottom. So this is depending upon this only the science, nanotechnology, everything started. So Feynman in 1959 talk is often cited as a source of inspiration for nanosciences, but it was virtually unknown outside to his small audience at the time and only published a scientific paper in 1992. This is the paper. So till then it was very much obscure. Nobody knew about this nanosciences, even that he told in 1959 for a small group of audience. So this understanding of nanosciences is Latin and Greek for me, because physics people usually go with this one because mainly this quantum mechanics is conceptual and challenging and requires mathematics knowledge, but it is an essential basis for understanding nanosciences. So who are interested can go to this field, quantum mechanics, and further go ahead. So what is this quantum mechanics is? The branch of mechanics that deals with the mathematical description of the motion and interaction of subatomic particles. So definitely this is Greek and Latin, so I don't want to go further in this one. So the term nanotechnology was coined by Taniguchi of the Tokyo Science University in 1974. He is generally used for materials with size between 1 to 100 nanometers. They were referred as nanoparticles, and any technology which uses these particles and these size of things for its behaviors is nanotechnology. So how big is a nanometer? You know now. So this is one of the important things that we should know. Day-to-day life, we know that water droplets won't stand on the leaf of the lotus. Why? This is because of, once again, the nanotechnology, or the concept of nano thing has emerged over there in the later parts of this time. Because on the leaf of the lotus, we have got micro goblets. Earlier we used to tell, but it is not micro, it is nano wax goblets, which will not allow the water droplet to stand like this or stand like this. The water droplet will be always like this, suggesting that it is super hydrophobic. Since it is the tank, it does not want the mud to stick on itself. That's why it keeps on cleaning. So this concept is explained by this nanosciences. So definition of nanotechnology is understanding and control of the matter at dimensions between approximately 1 to 100 nanometers, where unique phenomena enable novel applications. Encompassing nanoscale science, engineering and technology, nanotechnology involves imaging, measuring, modeling and manipulating the materials. These are the 4 important fields in which how to get it imaging, how to measure it. What is the importance of this measuring? Actually, it comes in our medicine, especially nanotechnological medicines, nanomedicines we call. We want to hit the target cell directly. Once if you take any tablet into your mouth, it will go to the gastrointestinal system where it disperses. The amount of absorption of that particular drug is around 30 to 40%. But if we give the same drug in the injection form, that to intramuscular, 20 to 30% will move. But if we give directly to the blood, that is the vein, then the amount of absorption is 100%. So in that case, when we want the drug to attack that particular target cell, we have to manufacture that particular medicine in such a way that these cells have got affinity towards this particular drug, which is of nanosize, and gets absorbed or attracted towards it, so that it acts upon that particular cell. So the cell is ready to suicide itself, because it itself is attracting the nano drug towards it. So that is how the technology is improved nowadays. Instead of taking that drug orally or in the injection form, we give it in target-oriented approach. So this is what asthma. Everybody, I think you people might be knowing about the inhalers. So forget about the inhalers. They won't use properly. They use as a scent also and go off. And they tell to the doctor that, see, in spite of using these inhalers so much, my asthma is not getting down. Okay, please tell me how you are using. They'll take that, shake it and put it like this. So this is what is happening. You know, what we want is nowadays it is coming as space inhalers, or just like the powder form. You just keep it and just smoke it. It will directly go to the lungs and act upon the terminal bronchioles. In asthma, the terminal bronchioles and the bronchioles will be squeezed because of the contraction of the smooth fibers present in these terminal bronchioles. This will be squeezed. So that's why that sound will come. When the person inhales, swing, sound will be coming during inhalation and exhalation. Do you remember that earlier we used to fire the firewood by using a blower? The sand pipe uttar the pipe sound kadmi. Sand pipe udre tra sound bartadi. That you have to appreciate those things. Then only. Similarly, when the bronchioles constrict, when the person inhales and exhales, then there will be a sound because of the passage of the air through a smaller diameter tube. So these drugs, when inhaled directly, then this causes dilatation of these constricted bronchioles so that the patient can breathe properly. If we tell nowadays that use these inhalers, but patients won't agree. Either use ಮಾಡ್ತಾಯಿದ್ರೆ ಜೀವನ ಪೂರ್ತಿ ಯೂಸ್ ಮಾಡಬೇಕು ಬೇಡ ನಮಗೆ ಗುಳಿಗೆ ಕೊಡಿ. So this is what is happening. Acceptance by the society for the change with respect to the improvement in science is inversely proportional in our society. We have to struggle for these things. We have to educate the people. What is this nanoscience? What is this nanotechnology? What is the advantage of it? Okay? So the evolution of nanotechnology is increasing day by day. Basic research, the applied research, then the early adaptation, rapid advancement, and eventually mass market. So our technologies will get enlightened only when there is markets. So earlier, nanotechnology is used in case of this computers, microchips, using batteries, small things. Everybody wanted the portability. They don't want to take any luggage. Once there was a time, even I have seen and witnessed that whenever the cricket is going to happen, they used to carry a transistor this much big with a belt over here to listen that commentary. But nowadays it has become the mobile. Simple thing. Whatever you want, you can do. At that time, when we told about these things, people used to laugh. But now it is a reality. You can immediately send any photo, any message, or any talk, or any lecture from here to America directly. If you see all these things, can't we believe that Arjuna used to fire the arrow by listening to the sound, Shabdavedi Vidya? How can you say it is false? Nowadays we are having radars. We have got radar system. Sitting here, I can blast another country. So this is the thing I wanted to tell you. We have got everything. Only thing is accepting. When somebody else tells regarding those things, we will accept. Otherwise, we won't accept this. Okay, this is also like this. when a particle of about 10 to the power of minus 9 meter is available, these are the things we can be done with that, and the utility can be done like that, means nobody used to believe. So research is required. I think you will go in this line. This is the one. Present areas of activities in the field of nanotechnology in India is, it is the agricultural field, biodefense, biotechnology, chemicals, communication, cosmetics, drug delivery, electronics, energy including solar cells, fertilizers, food technology, health systems, water purification system, metallurgy and materials, paints, pesticides, pharmaceuticals, security, textiles, all these things. So this nanoscience has got its wide opportunity. Only thing is, you people have to select where you have to go, in which field I have to work. So everything, every field of nanoscience has got its own importance. Only thing is, if we go and pursue in that particular line, we can definitely flourish. So what is this nanoparticle or nanomaterial? It is defined as, once again, any material that has unique or novel properties due to the nanoscale structure. It is any particle of about 10 to the power of minus 9 size, and its particles. That is that particle. It may be a crystal, or it may be a powder, or it may be tubes. So it can get any structure or any shape of that matter. See, nanoparticles can be clubbed together. This is diesel exhaust particle. This is carbon particle. This is a schematic representation of the diesel exhaust particle consisting of carbon core. This with potential harmful reactive transition metals and different organic chemicals. I am showing this slide because just to compare that what is the particular size of each and every particle. Speciality of nanoparticles is the size 10 to the power of minus 9 to 100 of that it can vary. It varies in reactivity, it varies in magnetism, thermally it varies, mechanically it varies, and optically it varies. So this particular particle at this size doesn't have similar properties. All the properties what we have learned, physical, chemical, structure, everything varies. And according to this variation, the property also changes and the utility of this particular particle also changes. You got my point? So at this size, all the properties what you have seen, what you have heard, for example, iron is attracted by the magnet in the big size. If you make it into powder, what happens? One is size, the surface area increases. The property varies. This is one. The nanoparticle size plays a key role in long circulation, biodistribution, and clearance. This is one of the important property which helps a doctor to treat. So this nanoparticle will be circulating in the body and it misses through the filtration of the kidney so that it cannot be thrown out of the body as early as possible. Even kidney also cannot recognize this particular particle as poisonous particle or friendly particle. Our glomerular filtration is not to that extent that it is not exposed. So maybe in next generation this change can occur because of mutation, as Sir was telling. So body has to accommodate for the particular environment. Even kidney has to sense the glomerular filtration should improve so that it can filter these particles and throw out of that body. And even the half-life concept, half-life of a particular particle for degradation purpose is also varies when the particle is in nano size. And liver, known for removal of all the toxic products in the body, cannot remove this. It will keep on storing itself, these particles in the liver, and ultimately, you know what happens when there is a scar. Something keeps on setting, fibrosis occurs, cells will be destroyed, and liver becomes fibrosed. That fibrosed condition we call it as cirrhosis. Cirrhosis of liver. So the main known cirrhosis concept is alcohol. Alcoholic cirrhosis is the most commonest thing nowadays. The next is silicosis. If the person is working in an asbestos or powdered exposed things, then definitely they will go for fibrosed liver. So these are all the particles which can prolong this biodistribution as well as this half-life periods. Variation in circulation in half-life, reduced response.
Sir, whether this reset conversion causes liver cirrhosis?
It can cause. It can cause. That we have also proved.
Liver cirrhosis.
Liver cirrhosis, the next stage. we showed that variation in the parameters of this bilirubin, direct and indirect ALKP phosphates. So the properties of the nanoparticles depends upon the tremendous force, driving force of diffusion, superparamagnetism, quantum confinement, semi-conduction, quantization of electrical charge, that's what. All the properties keeps on changing at this particular size. For example, even the color changes. The gold, as the size reduces, it can change its color. The normal color of the gold will be changed to red, blue when the particle reduces its size. Similarly, see silicon, this is insulator in bulk and in nanoscale it becomes a good conductor. And this copper, which is malleable and ductile in bulk, becomes stiff and elastic when it is in nanoscale. Titanium oxide, it is white in color, becomes colorless in nanoparticles. This is gold, chemically inert in bulk size and becomes chemically very active in case of nanoscale. So these things can occur. Things behave differently in nano world. Same continuation. Carbon in the form of graphite is soft in nanoscale, but in stronger and steel-like when it is 6 times lighter when it comes to these things. So similarly, this is copper, as I told you, gold. The nanoparticle size and shape dictates its rate of migration and ability to bind to the cell membrane. So what is the effect of shape? See, inside the blood vessel, in the blood, if there are nanoparticles circulating, why the deposition occurs? There is a stream of water running stream of water. If you throw some particles, mud, dust, leaves, or vanity bag, or a kerchief, how the turbulence acts upon these materials and push certain materials towards outside. Even though you throw all together as a bulk, they get separated because of the density of those bags or whatever material and the turbulence of the forces. So similarly here, spherical nanoparticles tend to remain in the center of the flow. Variable forces and torques exerted on the rod under flow allow them to migrate and drift towards the vessel wall, where they are able to bind to the wall receptors and extravasate through gaps between the cells of the endothelium. This is the concept explained behind atherosclerosis. Atherosclerosis is deposition of cholesterol into the blood vessels. So, blood vessel block agututte, sudden heart attack agututte, satonu. How does these coronary arteries only gets blocked? Aorta will be getting blocked. Or the plaques of atherosclerosis will be found in aorta only. Medium-sized arteries we call it. And in the peripheral arteries it is quite rare. I am not telling that it is because the turbulence of the blood flow in these blood vessels are very much more when compared to the peripheral blood. So that's why if any particle which is present over there tends to be pushed towards one side, especially these particles are present, cholesterol particles present in our body, which will be pushed towards the peripheries of the turbulence of the blood flow so that it gets deposited. Similarly, whenever there is coagulability disorders, the platelets will go on sticking. Platelets are round, whereas RBCs are discoid in shape. Even though WBCs are round, they are greater of size when compared to that of platelets. So platelets are pushed towards the periphery, they will go and deposit it on the walls, adhesion, and these further attracts these cholesterol or whatever particles towards it, resulting in dots, streaks, thickening, and even plaques, resulting in obstruction of the blood vessel. So we should not allow the cholesterol to enter into our body to prevent these things. Even though if you allow the cholesterol to enter into your body, you should be able to digest and break it by giving exercise to the body. So don't have junk things and don't sit idly. That's what I'm telling you. Okay? So these two are the pioneers. He is father of nanotechnology, a Nobel laureate in 1965, Richard Feynman, physicist, and he is the person who coined the word nanotechnology. The first ever concept was presented in 1959, as I told you. Invention of scanning tunneling microscope in 1981 and the discovery of fluorescence in 1985 led to the emergence of nanotechnology. The term technology was coined by this one. So he is the great person who told that there is plenty of room at the bottom. So this is the history of nanotechnology 2000 years back. sulfide nanocrystals were used by the Greeks and Romans to dye the hair. Nowadays also, everybody will go to the parlor and gets their streaks done. So once upon a time it was natural because of malnutrition. Whenever there is malnutrition of a particular person, we used to have flaxine on the hairs with multiple coloring. Because when the person is having good food, the growth was fine. When it is malnutrition, the color as well as the texture of the hair drops so that it comes as a different color. Once again, if he starts taking good food, a color comes. So these two things, if you compare, appears like a strip of flag. But nowadays it is difficult for us to find out in the ward or the outpatient whether it is a flaxine or whether it is artificially colored. People will say, you can look at the build. Okay, if the person is well built and well nourished, okay, he will find hefty and we can think that. And nowadays everybody are thinking of dieting and maintaining their figure at 0. So how can we think of whether it is malnutrition or artificially created malnutrition among the body? So that's why sir told me, you should enjoy. Just now, he told me. So this is what. And 1,000 years ago, gold nanoparticles of different sizes used to produce different colors and stained them. That's why Romans' windows are very much famous. The window glasses in Romans. The same concept is used in our Mysore palace also. 1959, 74, the word coined. 81, IBM developed scanning tunneling microscope. This is for analytical nanotechnology. 85, Blackmore, that is Buckley-Ball, scientist from the Rice University of Life. Then 86, engines were creations. So all these things, and recently even President Bush signed nanotechnology R&D Act for about 3.7 billion over 4 years. So I don't know what is the things going on in India, but it's not that much. We have got only in IIT Bombay which is going on certain works, but everything is towards the software.
Nanotechnology Institute.
Yes.
During Abdul Kalam period.
Yeah, he started in Mumbai, IIT. So methods of production of nanoparticles is nothing, very simple. It may be physical. One piece of China, what piece particle. The ultimate last part is the nanoparticle. Similarly, chemical. Take a chemical, do a chemical reaction so that it keeps on breaking. Maybe enzymes, classical examples. Enzymes, when used, they keep on breaking complicated substances into simpler substances. That is what we mean. And biological, that is green productions. So green synthesis of metals and metal oxides, nanoparticles have been highly attractive research area in the last decades. So this is because of the green revolution. This can open the career for you people. So try to look into this fact more and more. Numerous kinds of natural extracts have been employed as efficient resources for synthesis or fabrication of materials, both. So plant extract has been proven to possess high efficiency as stabilizing and reducing agents for synthesis of controlled materials. So at the end of the day, plant materials and extracts are the rich sources for nanoparticle production, and those, when it is labeled as plant extract, then it draws the attention of the market companies. So that's why this green revolution is supported by this biological synthesis of nanoparticles. So nanoanalysis is done by, this is itself is a huge branch. This is done by using different equipments like UV visible spectroscopy, particle size analyzers, ICP OES, then zeta potential analyzers, scanning tunneling microscope, electronic microscopes, all these things. So how to approach these things? Can go with this. So how this works? A nanoparticle functions with three principles. One is quantum physics, that is the electrical property and the optical property of the surface of the nanoparticle will vary with the size. So depending upon that, the activity starts. And surface area phenomena. A particle which is about, if you take a cube of about 1 centimeter into 1 centimeter, what is the overall total surface area of that? 6 square meters. So if you break it into 10 pieces, you will get 100, isn't it? If you, this whole thing is like this, if you cut like the bread piece slices and cut like this horizontally, vertically. Each block of about is 1 into 1 into 1 centimeter. If you take this whole thing as this one, so it increases by 100 times. Got it? So the surface area of these things increases. This is the simplest thing which we are using in even poisonous cases also. Activated charcoal is used. You know, whenever the person has taken consumed some poison, earlier we used to use, nowadays we are not using activated charcoal. It is nothing but a crystal of this charcoal which has got multiple facets and surface area is very much increased when it is given in the powdered granular form. We will make the patient to drink it, and since the surface is here more, it has got the capacity to absorb this poisonous material into that, and later it will not be digested and it will be excreted like that only along with the poisonous contents. So the surface area increases as the size decreases. So this is inverse proportion, and this cation exchange phenomenon occurs. Electromagnetic charges on particles originate by two means, that is the broken edges exposes negative charges on the surface. Isomorphic substitution, when a higher valent ion is replaced by a lower valent ion, the excess charge appears on the surface. So this variation in the charges, so all these things, these three principles help these nanoparticles to act in a different manner, in different circumstances, different temperature and different pressure. Got it? So these are all the different sources where we can use nanotechnology, energy, medicine drugs, nanotechnology, nano devices, optical engineering, defense and security, bioengineering, cosmetics, nano fields, all these things. So in electronics, nanotransistors, nanodiodes, and organic light emitting diodes, plasma displays, quantum computers, nanorobots. All these things have got the usage at a different particular fields. Nanorobots we use nowadays for the drug delivery system. These nanorobots are completely separable things. When it enters into the body with a particular drug, it reaches the particular target cells and releases the drugs over there. So application of nanotechnology in the energies, once again batteries, fuel cells, solar cells, in the materials, nanotubes, aerogels and nanoparticles. These nanotubes and aerogels are widely used in our orthopedic department, where if there is a non-union of a bone or a fracture of a bone, among the fractured sites we are going to use this paste, nanotube paste, for the generation of the bone tissue. If the bone tissue is not generated, it's okay. This is biocompatible nanogels which adheres these two bone fragments without using any intramedullary nail or external fixators or plates and screws. We can cure certain fractures by using these nanotubes gel. So similar way, targeted drug delivery, artificial retina is also nowadays used. Tissue regeneration, that's what I told you in the orthopedics. So these are the other nanotechnology applications in agriculture, mainly to improve the fertility of the things. We are using certain manures in this nanoparticle size. So in nanotechnology in India is IIT Mumbai is a premier organization in this field of technology. Research in the field of health, environment and medicine is still on. So starting in 2001, the government of India launched the Nanosciences and Initiative Technology. That is this one where you can approach this and you can approach IIT Mumbai for this, for mainly basic research promotion, infrastructure development of carrying out research, development of nanotechnologies and their application. This is the hot field nowadays. So madam wanted me to enlighten you people in these fields, that what is available in that, instead of going to the depth of the nanotechnology or nanotoxicology or nanosciences, what you people can achieve and go. So this is the one. Development of nanotechnologies and their application needs more and more manpower. You can go. And human resource development and international collaborations are already existing with these things. Apart from that, when we tell about certain good things, definitely there will be bad things. These two are like different faces of the same coin. So end of illness, medical advantages, these are the things. If we go to, this is one more important. See, recently, day before yesterday's Deccan Herald paper has given this article, nanopillars to toughen soft surfaces, burst bacteria. Day before yesterday's Deccan Herald, Madam told that there will be plenty of bacteria on a smooth surface or a towel also. So how to separate all these things in the genetic level she told. So similarly, earlier the house was used, the surface of the housing floor as well as the wall was very much rough, isn't it? Sagani soil, it used to be rough. Even if they leave it like that without plastering also, it was fine. Later plastering came, after that painting came, after that putty came. So we are doing putty on the wall to have a smooth feature surface. But what they are telling now here is this one. Because of the smooth surface, if the bacterial cell drops on the floor or it gets bombarded to the wall, it won't die, it without it seems. Because of the smoothness. So that's why they are starting this rough surface by technology known as nanopillars. So I suddenly thought, what is this nanopillar? These nanopillars are small, nano-sized pillars which are formed on these tiles. They want to make the surface rough, that's all. To be very simple. This walls also, they want to make it very rough. So this is what they are telling. Why the infections are more in hospitals when compared to outside? This we brand it as hospital-borne infections or nosocomial infections. Why? Because we launch bacteria of all sorts because of the patients who are coming in and out that hospital. We are not cleaning properly. That is the concept they are telling. We are keeping the hospitals very clean, but still infections is coming. Then start infection policy, they are telling that. What infection policy we can tell that? Nobody will take complete antibiotics in our country, including we doctors. If there is a course of about 5 days, once the symptoms come down, we will stop it. So, and you know what happens to these bacteria, they develop resistance. And ultimately they found it is a mega bug, it is a macro bug started, it is not resistant, it is not sensitive for any antibiotics. Now the bacteria are all becoming resistant. Why? Because of these things. So we want the hospital to be high-tech with soft flooring, soft smooth walls and all. So these bacteria go and stick according to this article over here. So that's why they want to change the concept. I don't know what marketing these people want to do now. Some minister might have got the tile factory where he want to change it and he want to give this to this. And we people, science people, should go and prove or disprove this. So this is what. So why I want to tell you this, nanopillar concept is coming up. Disadvantage, social, health and safety issues, social and political issues. See, wherever you go, this P doesn't leave us. Environmental issues, mainly with wars and grey goo. Actually, I didn't know what is this grey goo. So this is the nightmarish scenario of nanotechnology in which out-of-control self-replicating nanobots destroy the biosphere by endlessly producing replicas of themselves and feeding on the materials necessary for life. The term was coined by an American engineer, Eric Wexler, in his book. So have you seen that Rajnikant movie? Robot. Self-replicating fellow. So don't know, it can happen in future. So this is what he has told over here. And mass productions, loss of jobs, because once again, so oil-based becomes, diamonds become worthless, and atomic weapons more accessible and distant. So when nano today is only the beginning, it is just, at the end of the day, what we call it as the gist is nanoscience. What is nanoscience? What is nanomaterial? What we can do with these two things in future life. So the big thing is really small. I take another 10 minutes. Small is beautiful, but dangerous comes along with that. Small is really beautiful, but danger things comes along with it is the nanotoxicology. When these materials come in contact of the human body or the environment, it has got its own effects. These effects are natural and good for those particular particles, but they are not acceptable by our own body. So that we call it as adverse effects, and when that condition comes, it is nanotoxicology, deals with the toxic aspects of nanoparticles to the life and environment. Actually, this is my field. So we are starting with my field now, 10 minutes I did, that's all. what are the effects of these nanoparticles in the lives and environment is the work that we are conducting with respect to the health department, potential public health concern. So nowadays this potential public health concern is increasing day by day because we are all completely surrounded by these nanoparticles, resulting in undue adverse effects upon the human body, so that the government system cannot cope up with the change in the health scenario to produce 3 M's: the man, the money and the material. It is not giving properly. So that's why this is the thing which is going on nowadays. So which is classified into 2 types: biological toxicity and environmental toxicity. So what happens when these particles actually come in contact with a particular cell? Every organism on the earth has been exposed to the small and minute foreign particles by direct contact or indirect contact. Direct contact to the skin, indirect contact to the body is through inhalation or through ingestion. So when it enters the organ system of the human or the animals, it starts working at the cellular level by disrupting its process, interacting with the cell membranes, cell organelles, DNA, protein to establish a series of dynamic biophysico-chemical interactions. These biophysico-chemical interactions are not acceptable. Each and every interaction has got different signs and symptoms. And the response of these signs and symptoms for a particular drug is not equal in each and every human being. That's why we are called individuals. If I give paracetamol 500 mg for this girl, her fever can drop within 3 hours or 2 hours. If we give the same paracetamol to other, it won't drop even 24 hours also, because the response given by that particular body to that paracetamol molecule is not that efficient when compared to this one. So that is the individual. So each and every person on this earth has got its own individuality. He responds to this nanoparticle in a different manner. ನಾನು ಒಂದೇ ಎನ್ವಿರಾನ್ಮೆಂಟಲ್ಲಿ ಇದ್ದೀನಿ, ಅವಳು ಒಂದೇ ಎನ್ವಿರಾನ್ಮೆಂಟ್, ನನಗೆ ಮಾತ್ರ ರೋಗ, ಅವಳಿಗೆ ಮಾತ್ರ ಯಾಕೆ? ಅವ್ನು ಎಷ್ಟು ಸಿಗರೇಟ್ ಬೀಡಿ ಸೇಗ್ತಾನೆ ಸರ್, ಅವ್ನಿಗೆ ಏನೂ ಆಗಿಲ್ಲ, ನಾನು ಸೇದಿಲ್ಲ ಮಾಡಿಲ್ಲ, ನನಗೆ ಕೊಲೆಸ್ಟ್ರಾಲ್ ಅಂತೀರ. See, this is once again search topic, genes. So everything is nowadays genes. So whatever happens to a particular gene, then how the interaction takes place is also depending upon this. So all these health effects gain importance for different professionals when studied. See, the person can study the effect of these nanoparticles in general or gross health status, or using the organ system. What does this nanoparticle does for respiratory system? What does this nanoparticle does for cardiovascular system, nervous system? So like that, each and every organ system has got different projections towards the interaction between the cells of its own organ system and the nanoparticle. Got it? So, and effects upon the cells, chromosomes and DNA. This is your field. Core genetics field is this one. What does a nanoparticle form? What does a nanoparticle, how does a nanoparticle create hazard to this particular cell or genome or a DNA? That is your part, your cup of tea. So you should come across what should be done, what can be done at this time. At the end of the day, doctors are only using what you people give. So we are applied sciences, you are core science. So when a particle comes in contact with the brain, if the nanoparticles are very much more, it can cause neurological disorders, mainly two degenerative disorders: Parkinson's disease, Alzheimer's disease. Definitely there are certain materials which can cross the blood-brain barrier. And similarly, we have got thalidomide, one of the drug which is used as an anti-cancer. It can even cross the placental barrier and cause the fetus to failure in the generation or failure in the development of long bones. The mother who is taking thalidomide during the time of pregnancy, that child will lose both the lower limbs and upper limbs. You can see sequestrum of bone, small pieces of globules of bone tissues. They are not developed. Even sometimes the lips are also not developed. So these particles can cross the blood-brain barrier as well as the placental barriers also. Definitely it can. That's why the count has been decreased in the smoking study we did. So respiratory system, if you come across, you everybody know, starts from excessive water, sneezing, itching in the throat, that is rhinitis, sneezing, upper respiratory infections, pharyngitis, laryngitis. Then if it enters into the bronchus, it is bronchitis. Especially smokers who keep on smoking results in bronchitis. They keep on cough. If there is any secondary infection, then only sputum starts coming with yellowish color, thick. Otherwise, if it goes to the alveoli, then destruction of the alveoli, loss of elasticity of the alveoli resulting in broncholysis or bronchiectasis. So these are all the diseases which can come. And when it comes to the coronary, that is circulatory system, the deposition of these nanoparticles resulting in narrowing up of the blood vessels wall and increasing blood pressure. And if it deposits on the conduction system of the heart like SA nodes, then bundle of His, all these things, then conduction abnormality starts. That's why in chronic smokers, definitely people will go for conduction abnormalities. So these are all the things. And diseases of the kidney, diseases of the lymphatics, skin. So all these things will be established. So when the particles gets inspired by the lung, from the lung it can go for, it can cause damage in the lungs, or it can cause autoregulation and acts upon the blood vessels, causing variation in the diameter by shrinking them or dilating them. When the blood vessel gets dilated, the blood pressure decreases. When the blood vessel shrinks, the blood pressure increases. So unknowing causes of these increased or decreased blood pressures. We have to tackle all these things. And the third one is particle translocation, going from one particle to the other area. This can, if it comes to the genetic level, then translocation of these genes can causing various diseases and disorders or syndromes depending upon that particular site. That is same thing, particles depositing, and it triggers the oxidative stress. So this is what you can see the clots. I don't know this picture is not. See, this is the atherosclerotic plaque. The whole aorta is completely studded with the atheromas. This is pale yellow. This is the aorta normal wall. See, this yellow is completely, it is the deposition of the fat. And you can see this blackish things over here. This is the normal color, or red is the normal color. You can see these black dots, black streaks, black patches. All these are carbon things. See here, completely clogged. If you feel that during the postpartum examination, the black thing, we can feel like just sudden color signal. And to confirm that, we just throw it on the table. Sound, so it has become granulated completely. So this is the main cause, especially Namgal lung system. We are exposed to, almost 50 years I have exposed. You people 20-25 years have been exposed to this environment. Definitely after 3 to 4 years, these carbon particles gets deposited in the body.
They won't come out, sir.
They can't disperse, they can't digest. They just be like that. ಕಾಲು ಮುಚ್ಚಿಟ್ಕೊಂಡ್ಬಿಟ್ಟಿರುತ್ತೆ, ತೆಗೆದಿರಲ್ಲ, ಏನಾಗುತ್ತೆ ಸುತ್ತ ಫೈಬ್ರೋಸಿಸ್ ಆಗ್ಬಿಟ್ಟು ಮುಳ್ಳು ಒಳಗೆ ಇರುತ್ತೆ. ಸಮಟೈಮ್ಸ್ ಸೆಕೆಂಡರಿ ಇನ್ಫೆಕ್ಷನ್ ಆಯಿತು ಅಂದರೆ ಕ್ಯೂ ಬರುತ್ತೆ. ಕ್ಯೂ ಬಂದಾಗ ಹಿಂಗಂತೂ ಬಂತು ಸರ್ ಮುಳ್ಳು ಅಂತ. ಸೊ ಇಟ್ ವಿಲ್ ಬಿ ಲೈಕ್ ದಟ್. ಸಣ್ಣ ಮುಳ್ಳು ಚುಚ್ಕೊಂಡಾಗ ಪಿನ್ ಚುಚ್ಚಿ ತೆಗಿತಾರೆ. ತೆಗಿಯಕ್ಕೆ ಬರ್ದಿದ್ರೆ ಏನು ಮಾಡ್ತಾರೆ? ಬಿಡ ಆ ಕಡೆ ಹೋಯ್ತು ಬಂತು ಎರಡೇದಾಗ. ಕೆಲವು ಫೈಬ್ರೋಸಿಸ್ ಆಗಿ ಗ್ರ್ಯಾನ್ಯುಲೋಮಾ ಆಗ್ಬಿಟ್ಟು ಅಲ್ಲೇ ಉಳ್ಕೊಳ್ಳುತ್ತೆ. ಇಫ್ ಯು ಎಕ್ಸೈಸ್ ದಟ್ ಗ್ರ್ಯಾನ್ಯುಲೋಮಾ ಕಟ್ ಓಪನ್ ಅಂಡ್ ಸಿ ಯು ಕ್ಯಾನ್ ಸಿ ದಟ್ ಥಾರ್ನ್ ಪಾರ್ಟಿಕಲ್ over there inside. Otherwise, whenever that whole thing gets infected, earlier what they used to do, ಚೆನ್ನಾಗಿ ಚುಚ್ಚಿಬಿಟ್ಟು ಗಾಯ ಮಾಡಿಬಿಟ್ಟು ಈ ಹತ್ತಿ ಹಾಲು ಯಾವುದೋ ಒಂದು ಹಾಲು ಕಟ್ಬಿಡ್ತಾ ಇದ್ರು. ಇಟ್ ಯೂಸ್ ಟು ಗೋ ಸೆಕೆಂಡರಿ ಇನ್ಫೆಕ್ಷನ್ ಆ್ಯಂಡ್ ಸುತ್ತ ಕೊಳ್ತೋಗ್ಬಿಟ್ಟು ಆಚೆ ಬರ್ತಾ ಇತ್ತು. ಅದು ಪೂರ್ತಿ ಆಚೆ ಬಂದಮೇಲೆ ಗ್ರ್ಯಾನ್ಯುಲೇಷನ್ ಟಿಶ್ಯೂ ಡೆವಲಪ್ ಆಗ್ಬಿಟ್ಟು ಗಾಯ ವಾಸಿ ಹೋಗ್ತಾ ಇತ್ತು. ಇಟ್ ಯೂಸ್ ಟು ಟೇಕ್ 1 month. ಸೊ ಮುಳ್ಳು ಬಂತು ಅಂತ ದೇ ಯೂಸ್ ಟು ಗೆಟ್ ಸ್ಯಾಟಿಸ್ಫೈಡ್. ದಿಸ್ ಇಸ್ ವಾಟ್ ಇಸ್ ಹ್ಯಾಪನಿಂಗ್. So this is once again the deposition of these atherosclerotic plaques in this experimented mice, that is repeated pulmonary exposure to diesel exhaust particles increases size of these plaques. This has been studied. So common nanotoxicity is occurring mainly due to air pollution. This air pollution is mainly by 3 things. One is automobiles. Munche vehicle, how many vehicles are getting released onto the road? Tell me. ಮುಂಚೆ ಒಂದು ಮನೆಯಲ್ಲಿ ಒಂದು ವೆಹಿಕಲ್ ಇದ್ದರೆ ಅವರೇ ದೊಡ್ಡ ಶ್ರೀಮಂತರು. ಈಗ ಒಂದು ಮನೆಯಲ್ಲಿ 4 ಬೈಕು 3 ಕಾರ್ ಇರ್ತವೆ. ಪಕ್ಕದ ಬೀದಿಗೆ ಹಾಲು ತರಕ್ಕೆ ಹೋಗೋಕ್ಕೂ ಫುಲ್ ಸಿಂಗಾರ ಮಾಡ್ಕೊಂಡು ಬೈಕ್ ಕೂತ್ಕೊಳ್ಳೋದು ಅಷ್ಟು ಹೊಗೆ ಬರುತ್ತೆ. ಸಿ ಯು ಪೀಪಲ್ ಪ್ಲೀಸ್ ಪ್ರಾಕ್ಟೀಸ್ not to use these vehicles, at least in this green campus. Allow this to be green. Walk. Unnecessarily you are going to join this aerobics, giving 10,000 bucks per year, and you want to go for gym by paying 10,000 bucks once again. And nowadays the apps have come. Walking 10,000 steps per day. Everybody will be keeping on his mobile watch and
these medical servicing centers, especially Activa and other, they will be so narrow and non-aerated, and every
that's what my PhD topic now. Exposure to these things.
Balance Honda or any other, the servicing area will be so compact and totally not aerated.
ಅದು ethical clearance ಅಲ್ಲಿ ಬಹಳ ಪ್ರಾಬ್ಲಮ್ ಆಗಿತ್ತು ಮೇಡಮ್.
ಆಮೇಲೆ ಇನ್ನೂ ಒಂದು ಸಮಸ್ಯೆ ಏನಂದ್ರೆ these temples, the ಗರ್ಭಗುಡಿನಲ್ಲಿ ಎಲ್ಲೂ ವೆಂಟಿಲೇಷನ್ ಇಲ್ಲ, and the whole
carbon particles
camphor and the other things. I think one study we have to do regarding deposition of carbon in these slums. They'll be exposed, they'll be burning.
Hello, what we have to do after doing that is the problem. See, you people can create a change. Let us not use these vehicles. Let us not go to these aerobics or gym centers which are cropping up like mushrooms, isn't it? So this is what is happening. Automobile exhaust and factory exhaust and field fires. ಸಿಕ್ಕ ಕಡೆ ಬೆಂಕಿ ಹಾಕ್ತಾರೆ. ಈಗಂತೂ 4 ಹೆಜ್ಜೆಗೂ ಒಬ್ಬ ಕ್ಯಾಂಟೀನ್ ಇಟ್ಕೊತಾನೆ, ಪ್ಲಾಸ್ಟಿಕ್ ಲೋಟ ಇಟ್ಕೊತಾನೆ, ಸಾಯಂಕಾಲ ಹೋಗ್ತಾ ಬೆಂಕಿ ಹಾಕ್ಬಿಟ್ಟು ಹೋಗ್ತಾರೆ. These are carcinogenics. These are real carcinogenics. If there is anything like that, immediately you curb it. ನಾವು ಹೇಳಿದ ಜಗಳ ಆಡೋಕ್ಕಾಗಲ್ಲ, ಹೇಳ್ತೀವಿ ಆಫ್ ಮಾಡೋ ಹಚ್ಬೇಡ ಅಂತ, ನಿಂಗೆ ಹೋಗಲಿ ಅಂತ ನಿಜವಲ್ಲ. ಸೊ ಅಟ್ಲೀಸ್ಟ್ ನೀವು ಹುಡುಗರು 4 ಜನ ಸೇರಿಕೊಂಡು ಒಡೆಯಕಾಲ ಹೋಗ್ಬೋದು. ಕ್ರಾಸ್ ಆಫ್ ಮಾಡ್ತೇವೆ ಇಲ್ವ ನಮ್ಮ ಅಂತ ನೀವು ಸ್ಟಾರ್ಟ್ ಮಾಡಬೇಕು. Otherwise, you can call this corporation, one number is there. You can call this corporation and tell that, sir, in unnecessary ಬೆಂಕಿ ಹಾಕ್ತಿದ್ದಾರೆ, it is creating hazard and havoc in our environment ಅಂತ ಅಂದರೆ ಅವ್ರು ಬಂದು ಆಫ್ ಮಾಡಿಸ್ತಾರೆ. ಆ್ಯಕ್ಚುಲಿ ಕೇಸು ಹಾಕ್ಬೋದು, ಹಚ್ಚಿರೋರು ಗೊತ್ತಿದೆ. So this is a system in controlling this fires. Actually field fire ಪಂಜಾಬಲ್ಲಿ ಶುಗರ್ ಕೇನ್ ಬೆಳೀತಾರೆ, ಕಬ್ಬು ಬೆಳೆದ ಮೇಲೆ ಏನೋ ಕೀಳಲ್ಲ, ಇಲ್ಲ ಉಳಬೇಕು ಉಳಲ್ಲ, ಎರಡೂ ಮಾಡಲ್ಲ. ಹಾಗೆ ಉದ್ಬಿಟ್ಟು ಬಿಟ್ಟರೆ ಒಳ್ಳೆ ಗೊಬ್ಬರ ಆಗುತ್ತೆ, ಬೆಂಕಿ ಹಚ್ಬಿಡುತ್ತೆ. ನಮ್ಮ ಕಡೆನೇ ನೋಡಿದಿಲ್ಲ, ಅದಕ್ಕೆ ಬೆಂಕಿ ಹಚ್ಬಿಡ್ತಾರೆ ಕಟಾವಾದ್ಮೇಲೆ. ಸೊ ವಾಟ್ ಹ್ಯಾಪನ್ಸ್ ಟು ದೀಸ್ ಬ್ಲ್ಯಾಕ್ ಕೋರ್ ಸ್ಮೋಕ್ which spreads everywhere. This was the first and the foremost cause of air pollution in Delhi 10 years ago. And they are simply blaming our crackers also. Nowadays they started green crackers this time. So I don't know how far it is safe with these things. So these field fires, factory exhaust and automobile exhaust has to be reduced in the society. And the color of the exhaust determines the health of the car also. See, cylinder head gasket is damaged. Cooling fluid enters into the combustion chamber. This color will be coming. And the smell will be typical. Cars is likely to burn engine oil, this type of thick smoke. If it is black, burning gas oil over the car engine, carbon rich. If you go behind KSRTC bus, you can get a typical smell, and even if you take it like this, the powder of carbon will be deposited, especially when you are wearing a white shirt, a white dress, you can see that. ಈ ಥರ ಕಾರ್ ಮುಂದೆ ಹೋಗ್ತಾ ಇದ್ದಾರೆ, ಟ್ರಾಫಿಕ್ ಪೊಲೀಸರು ಬಿಟ್ಬಿಡ್ತಾನೆ. ಇಲ್ಲಿ ಯಾರೋ ಇಬ್ಬರು ಕಪಲ್ಸ್ ನೀಟಾಗಿ ಒಂದು ರೌಂಡ್ ಹೋಗೋಣ ಅಂತ ಹಿಂದ್ಗಡೆ ಹೆಲ್ಮೆಟ್ ಹಾಕಿಲ್ಲ ಅಂದರೆ ಮರದ ಸಂದಿಯಿಂದ ಹಾಲು ಹಿಡ್ಕೊತಾರೆ. ಹೆಲ್ಮೆಟ್ ಹಾಕಿಲ್ಲ ಅಂತ ಈಗ ನಂಬರ್ ಪ್ಲೇಟ್ಗಳು ಸ್ಟಾರ್ಟ್ ಆಗ್ತದೆ, ಗೊತ್ತಲ್ಲ ಅದು? ವಿ ಆರ್ ಸಪೋಸ್ ಟು ಚೇಂಜ್ ದ ನಂಬರ್ ಪ್ಲೇಟ್ಸ್ ಆಫ್ ಅವರ್ ವೆಹಿಕಲ್ಸ್. ಸೊ 17th was the deadline. Yesterday they have extended it to February. So a car, a vehicle, a lorry will be going like this. They think that this is very healthy. Not wearing the helmet by a pillion rider or a child who is sitting in front on the tank is a big mistake. So this is what our country is doing. So whenever the carbon particle exhaust, this is the carbon particle with multiple deposits of heavy metals on that. This is known as particulate matter. This particulate matter gets inhaled when it is exposed. When our body gets exposed, this enters through contact, inhalation or ingestion, and the action starts by disrupting the cell in such a way resulting in variation in the hemogram, hepatofunctions, renal functions, and oxidative stress markers and total antioxidant capacity varies. Cytogenetic and molecular changes at the DNA level occurs, and even the change occurs at the smaller group, that is, this is one of the studies done over here. So the results are the diseases and the disorders, and the mainly thing is primary prevention, early detection of the occupational hazards, and the air pollution has to be decreased. So when a nanoparticle enters into the body, this is the commonest mechanism. Inflammatory response is triggered, and cell insult occurs, changes in the basic metabolism and cell protective mechanism and generative of oxidative stress. And at the cellular level or inside the cell, these are all the changes occurs. So you can take up the study in this genotoxicity. What is this nanoparticle does to the particular cell and its core things. So when we did a study, when compared with the normal of 500 and then exposed controlled is 300 and exposed is 500. These people are exposed to the diesel combustion nanoparticles 8 hours continuously in a day for the past 8 years, and without wearing any mask or protective mechanism. This was the changes we could find. Classically, the eosinophils has been increased, and similarly, this is the most important thing. That's why whenever you get exposed to any dusty weather, immediately you start sneezing and excessive watery raining. That is, mast cells start its activity, resulting in excessive secretion to throw that particle away from the body. This goes and sticks into the mucosal cells, irritating the mucosal cell. ಒಬ್ಬ ಪಕ್ಕದಲ್ಲಿ ಕೂತ್ಕೊಂಡು ಸುಮ್ಮನೆ ಅಂತ ಇದ್ರೆ ಸ್ಟಾರ್ಟಿಂಗ್ ಒಂದು 5 ನಿಮಿಷ ಚೆನ್ನಾಗಿರುತ್ತೆ, ಆಮೇಲೆ ಇದು ಬಿಡೋದು ಅನ್ನಲ್ಲ. So nowadays we have got separate chairs. We used to sit on a bench even in our MBBS. Their same thing. Later they did 4 people per bench. So this is what happens. If this nanoparticle goes and sits on a cell and starts irritating, what happens? Excessive secretion of that particular cell, especially mucosa, resulting in rhinitis. Then irritation causes the nerve-ending ticklings, resulting in sneezing. The body tries to throw that particle away from, starts sneezing. So these are all the things. Next, if we go to the liver, that's what ma'am was telling. When we did this study, see, all the parameters in the liver has been increased with this p-value, significant p-value. All the liver parameters were increased. And when it became for this one, kidney functions, once again, see, everything is increased. So blood. And when we came to the oxidative stress markers, the salt, catalase, MDAs and TAC, all the things were increased. So this shows active inflammation is going on in that particular person. Even though he does not complain of any disease or any signs and symptoms, the inflammatory process in his body who is getting exposed this particular particle will be always in progress, so that one fine day he may develop a degenerative disorder. It may be Parkinson's, or it may be Alzheimer's, or it may be glomerular toxicity resulting in adult type of nephrotic syndrome. You might have heard about nephrotic syndrome. This is a disease usually used to come in childhood, but nowadays we are seeing more and more cases of nephrotic syndrome in adults. So we brand it as adult nephrotic syndrome, where suddenly a lady who is sleeping, urine output decreases, the facial puffiness starts. If we see the urinary protein, it is very much excreted, and the blood protein, that is the plasma protein level, has been drastically decreased because of this inflammatory process. So these are all the chronic things which are occurring. When it comes to chromosomal level, aberrations we are seeing, like deletions, breaks, dicentrics, satellite absence and elongated, all these things in particular chromosomes. So, this was an explanation given in brief. So, nanottoxicology, a particular nanoparticle can affect each and every organ system, and there may be immediate effects upon that, or it may be a chronic effect resulting in a disease in a later aspect of this. So nanotoxicology is a branch of bio-nanoscience. We introduced one more word, bio-nanoscience, which deals with the study and application of toxicity of nanomaterials. Nanotoxicological studies are intended to determine at what extent their properties may pose a threat to the environment and the human beings. So definitely you can also opt this field, that what are these nanoparticles effect upon the live cells. It may be animals or humans. Thank you so much for your attention.
If there are any questions, you can ask us. Sir, I have one question. So, there are constant use of systemic pesticides and fungicides in agricultural fields, right? And there is also use of nanoparticles. So, how far would it take to affect the patient, like whoever is spraying it? Like most of them use foliar method to spray them, and it's very rare they do a basic application, right? So, what is the percentage of getting affected with the asthma with such farmers, and how can it be stopped?
If you come to the asthma, it is almost, when you come to asthma in particular, it is almost 90 to 95%. If 10 farmers are spraying today to their field of about 1 acre, they are going to use about minimum 3 to 4 barrels on that with dilution, of course. But it also depends upon the wind breeze also. If he is spraying and the wind breeze goes like that, it's fine. He is cleared on that day. Otherwise, if there is no wind breeze or wind breeze is towards him, definitely he will have giddiness and he will have sneezing, irritation of the throat as well as skin within 3 to 6 hours, and he will come to the hospital. If you sit in the casualty for about 2 hours, you will come to it. So, triggering of asthma is almost 90 to 95% by 12 to 24 hours, number one. And number two, definitely we have got so many cases that to such an extent there is dermatitis, that is pesticide dermatitis, or simple thing, arasan soap, arasan and soap. So that has caused so much chemical irritation to the particular skin, yen haakudru hogala. Talakat kade yav soap arasan soap, arasan soap, arasan soap. Arasan and so don't use that soap, automatically it vanished. So this is the contact thing. So whenever we are using these pesticides, definitely we have to cover and follow the protocol. But our farmers are not that much rich to use these cosmetics to close them or use these kits to cover themselves. Definitely they will have, and that is why more and more amount of degenerative diseases are increasing, especially even diabetes, one of the heterogenic causes. We will tell the amount of diabetes as well as hypertension, coronary heart disease is increasing. One of the causes is exposure to these pesticides. Earlier we used to tell that smoking and alcohol were the risk factors. Nowadays even this has been added, exposure to this. If you have time, I can quote an example. See, recently in Tamil Nadu, about 15 to 20 children suddenly started itching on the body, vomiting, nausea. It's not controlled by our routine treatment. Then what is happening? Okay, generally we manage the case and ultimately all the 15, 16 students were fine. Food examination was done, room examination was done, everything was normal. We couldn't find any of the cause of that particular hostel, why these 15, 20 children suffered from this particular disease, which did not respond to our routine treatment. We were supposed to manage these cases for about 3 to 5 days hospitalization. The same thing was repeated after 15-20 days for the same group of children. Then immediately we started thinking, what is happening? Alien aitu hostel here. Welcome to the point. Mass agi batte hogutade. ಬೆಡ್ಶೀಟ್ಸು ಆ ಮಕ್ಕಳ ಬಟ್ಟೆ ಎವ್ರಿಥಿಂಗ್ ಕವರ್ಡ್. ಸೊ ಹೋಗದ ಮೇಲೆ ಎಲ್ಲಿ ಒಣಗಾಕಿದ್ರು ಅಂತಂದರೆ ದೇರ್ ವಾಸ್ ಎ ಫೀಲ್ಡ್ ವಿಚ್ ವಾಸ್ ಸ್ಪ್ರೇಡ್ ಬೈ ಆರ್ಗನೋಫಾಸ್ಫರಸ್ ಪೆಸ್ಟಿಸೈಡ್. ಆನ್ ದಟ್ ದೇ ಯೂಸ್ ಟು ಗೋ ಅಂಡ್ ಕೀಪ್ ದೀಸ್ clothes for drying. So these people used to wear this and they started these problems. And the clothings were taken for forensic analysis, and then the carbon particle, apart from this organophosphorus particles, were so high that it can cause these problems. So later, this was the conclusion which came. So even though when it is wet clothes, if you keep it on the plants or crops which are sprayed with this organophosphorus or whatever pesticides are she told, definitely these things will get absorbed and transferred transdermally. Through the skin it can get transferred. And in the next episode, when such thing happened, then immediately pseudocholinesterase, one of the markers in case of organophosphorus pesticide poisoning, were analyzed, and these pseudocholinesterase enzyme was elevated in all the 15 children. So this gave a conclusion. So similarly, when we use pesticides without anything, 90 to 95% definitely there will be ill effects. It may be conjunctival ill effects, or it may be skin, or it may be generalized nausea and vomiting. First symptoms start. So it happens.
I would like to extend my heartfelt thanks to Dr.Mahadeshwara Prasad for a very informative lecture on nanotoxicology, which is entirely a different topic. And not only he has enlightened us with that, he has also shed light on various other topics associated with nanotoxicology that we encounter on a daily basis. Thanks again, sir. I request our chairperson to felicitate Dr. Mahadeshwara Prasad.
As sir also, this is my home.
Shivanandappa sir.
Thank you everyone for your presence and time.